You're a breakout session leader and doing a report back. Can you come to the front? Please, thank you. We'll go in order of the breakout sessions. So regenerative medicine, I think, was first. Is that right? No. Genetic diversity with Daniel and Aravinda, right? Okay, so they are coming in virtually.
Okay, great. If they're coming in virtually, then presumably we are all set to go. So the first breakout session was genetic diseases and genomic risk factors with Aravinda Chakravarti and Daniel MacArthur. So over to you guys.
Yours?
Thank you very much. Yes, please do get started.
Well, hopefully they can.
Before of us.
Can you hear us in the hall?
Yes,
Would somebody wave?
Yes, good, we can hear you.
Okay. Good morning.
Excellent. Thank you.
Daniel.
All right, fantastic.
Sorry,
I'll just share my screen, 1 second.
You cannot hear us unless we speak to a mic. So we'll have a mic on our side.
Yes, I know, I'm speaking.
All right, there's no mic on your side. Excellent. Okay, perfect. I'll get talking. So we had a great session. Thanks again to Aravinda for doing the bulk of the moderation to our discussants and everyone who helped out in the room. I just wanted to touch on a few points from the session, acknowledging that there's much useful discussion that can't be captured in a 5-minute summary. We focused heavily on the various ways in which single-cell RNA-seq and other multi-omic data could be integrated at scale, ideally with genotype data to inform on disease mechanisms. One of the major themes from the session was about the value of scale. We discussed the fact that at the moment there are ongoing studies with single-cell RNA and genotypes of various forms in the high hundreds, reaching up into the thousands of population samples. These data, although they don't necessarily directly involve disease patients, can be integrated with data like GWAS to investigate the mechanisms of disease. It will take longer for samples from diseased patients to reach large numbers, but of course the small sample sizes currently available is still extremely useful for understanding mechanism, especially in the context of rare disease with variants with very large effects. There was some discussion about the value of calling genetic variants directly from single-cell RNA-seq data, including from 3' tag data. It's worth noting, although that's possible, it's very difficult to use that in the context of expression QTL discovery studies. Ideally, we'd want genotype or preferably even whole genome sequencing data from all samples wherever possible as a default. There was some discussion about the need for easy, robust tools for study design and power estimates, accommodating things like the distribution of effect sizes, the distribution of the rarity of individual cell types, and the numbers of samples that can be included. And of course, an acknowledgement that blood will be the first tissue to really achieve massive scale because of its easy accessibility, but understanding the mechanisms of disease will obviously require accessing data from many different tissues. A large chunk of the session then followed on around multiple different ways to address that final point. So looking at tissues that are relevant to disease. First area of discussion was around integrating tissues from disease patients. There's obviously many studies currently underway in this space. Tissue accessibility, though, is a major challenge for many diseases, particularly those affecting specific developmental cell types or tissues that are really difficult to access in living individuals. There's no easy answers for many of these. This will just be intrinsically difficult. There was a lengthy discussion about the care needed to distinguish primary causal effects of genetic variation from the secondary, which could either be compensatory or pathological changes that are caused by the disease process itself. There are various ways to untangle those primary and secondary effects, including, for instance, time course studies across the disease state, either in patients or in cell models of disease, or statistical inference methods, and Mendelian randomization was mentioned here, although here it's worth acknowledging the need for large N for tools like MR to be well powered. We also talked about the role of induced pluripotent stem cells and other types of differentiable cell models. iPSCs are obviously expensive at scale, so in order for these to work at large scale, we need to think about ways to reduce cost. such as, for instance, accepting polyclonality of cells or carefully selecting differentiation pathways that we go down. We can't explore the entire differentiation space. And one point here that I raised and others mentioned as well is that it's not necessarily the case that we need to have a really high fidelity model of individual tissues in the sense that they perfectly reflect in situ biology. It may be enough that we have a large number of different differentiation pathways that allow us to explore the space of gene expression and cellular phenotype, with each one of those not necessarily perfectly reflecting an actual cell type that we might see in biology. There was a really interesting discussion, I thought, about the different ways that statistical geneticists and cellular biologists can approach problems, the need for multidisciplinarity in approaching these questions. One particular mention was about statistical geneticists often focusing on genes as an outcome measure, whereas for cellular biologists, often the measure of interest is a higher order phenotype, such as a gene expression program or higher order cell phenotypes. And we need to think about how we can integrate these different approaches to make sense of the underlying biology. Lots of other topics were covered, including computational modeling, multiomics, and other things that I don't have time to cover today. So I'm going to stop there. Aravinda, was there anything that I missed that you'd like to mention?
No, the only other thing, I think many participants brought this up, is that even though studies of human disease are important, the broad goal was to understand biology broadly. And it could be humans, and of course, there are many other kinds of models that could be used, model systems. Clearly, the mouse is being used, as well as other kinds of models, such as, iPSCs are one, but clearly the others could be used.
Excellent. And I'll stop there. Thanks very much.
Great, thank you very much indeed. So what I didn't say at the beginning is that we're going to have 5 minutes presentations from each of the speakers, and then we'll take questions at the end. So hopefully you'll be able to stay online, and then we'll direct questions your way when they come. So we'll now move over to the second session, which was regenerative medicine. So if I could ask Henry Jasper and Barbara Treitlein to give their update.
All right, so in the regenerative medicine session, we focused on the challenges and limitations in regenerative medicine with relation to both in vivo reprogramming, understanding regenerative processes in vivo, perturbing and modulating regenerative processes in vivo, as well as cell therapies potentially. And of course, the discussion was about which data sets do we need, what are the limitations, the technological limitations that we need to overcome in order to really understand regeneration as a process, and to learn how to perturb it and how to predict cell states in the long run. Next slide, please.
Yes, so here's a summary of all the things we discussed. And so overall, there was a major topic being the in vitro engineered cells and tissues. So this being organoids or reprogrammed cells or in vitro differentiated cells and their use for transplantation. And so one important aspect that we discussed was actually being sure what you want to transplant at the end is what you think you transplant, and how to assess what the cell is and how similar it is to the in vivo counterpart. And we discussed that we are currently still not yet there to really have a good computational measurement or to quantify similarities between in vitro and in vivo. We do correlations, we say they are that percentage similar, but to really identify whether the cell is the same or what is still missing needs further developments on the computational side. And we also discussed that we need benchmarking tools, benchmarking of computational tools that are robust and reproducible for the clinic, such that when we have cells that are differentiated or tissues differentiated in vitro, that we know, are they good enough to be transplanted? And So, and there we went a lot into the discussion of different modalities we can profile with the existing techniques we have, and we discussed that we still need to learn what modality is the most informative and what aspect of that modality. So how can we, what do we need to measure in order to make sure we have the right cell that after transplantation regenerates the tissue and doesn't go off into unwanted, undesired cell types or states and potentially lead to cancer. So we discussed a lot about the different modalities and how they actually, how they inform us about cell states. We also discussed here that actually it is not enough to know the transcriptome of a cell alone, because it could be that once it's transplanted in the tissue, in that microenvironment, in that niche, it goes off into a new direction and generate new states that you don't want. So we said that in addition to just profiling the cells, we also need to perturb them and understand the perturbation landscape of the cells to really know is it doing the things we want it to do once we transplant it into the microenvironment. Then we discussed a lot about characterization of regeneration processes, and of course in this case we have a lot of model systems available, a lot of mouse models and other animal models to study regeneration, and they are potentially a good proxy for the human counterpart. And so we need these model systems from really highly regenerative species like Planaria, axolotl, hydra, and so on, through zebrafish and so on, to mouse and chicken and rabbit, and to understand when we injure a tissue what happens, and we can do really fine time course measurements. Also, in vitro models are very important. In this case, we have a modular system where we can add a certain cell type to the system, we can leave it out and see how cells respond, and we can perturb it. You saw this all for example, in Prisca Liberali's talk. And of course, we also need to look at human tissues, and in that case, we are limited with what samples we have available, and we just need to, in this case, profile what we can get. And so generally we said, data is important, so it's definitely important to generate more data on all kinds of regeneration contexts. But then there was the question, can we, from all these atlases and data that is there, predict what is the right cell state, and how do I get to that right cell state? And so we discussed, for example, the Yamanaka factors. Can we, from existing data, predict these are the Yamanaka factors that we need to use? And we said we are not yet there. We need to also, again, work more on the computation. And then we had two more points I just briefly mentioned: genetic variability that can really affect the success of regenerative therapies, and we need to understand what's the impact of genetic variability, and regeneration versus cancer. So how can I ensure a really successful regeneration of a tissue without the cells going actually off into formation of cancer? And this is a problem for in vitro tissue, but also for making tissue-resident stem cells regenerate the tissue if they have somatic mutations acquired.
Brilliant, thank you very much indeed. And now we've got the cell-based immunotherapy, Dieter Lambrechts and Antonia Müller. Can we have the first slide? Send them by email? Yeah, do you need the slides, or can you just start talking about it? You'd rather wait for the slide? Yeah, okay.
Depends on if they have it quickly or...
Yeah. Otherwise, we could go on to the fourth, which was the precision oncology, but I really need to be guided here by the tech people to tell me what works best.
right, thank you. There is the slide. So we are the cell-based immunotherapy group, and we had a very interesting discussion. I come more from a clinical and translational side. Here's a cancer researcher. We first discussed the key problems that we identified in cellular therapies, and we focused on hematopoietic stem cell transplantation and immune effector cell therapies. And we said one key problem here is that the cells that we give are a very heterogeneous mixture of cells that are typically in the clinical setting widely used but totally poorly characterized. And this will influence variable therapy responses that we also see in the clinic. We have all the data, and those responses are also poorly controllable. Like, yeah, we give the whole shish kebab of cells, and then we wait and see what happens. Also, the side effects limit the broad applicability of those cell-based therapies. An allotransplant could be used to cure autoimmune diseases and also some metabolic disorders, but the high rate of morbidity and mortality coming along with those therapies prevents a broader applicability. Same accounts for immune effector cell therapies as CAR T cells. So what gives rise to these side effects? Again, probably partly the heterogeneity of delivered immune cells that could trigger side effects. And in addition then, in the context of CAR T cells, for example, on target off-tumor effects. This is a very important topic. Then finally, another problem identified is post-infusion in vivo dynamics and function of transplanted cells that need to be elucidated.
Okay, thank you, Antonia. So how can the Human Cell Atlas contribute to this? Well, this is briefly summarized in the next 4 points. So first thing is that we think that a curated database of immune cell types and states across healthy tissue at different ages and with different comorbidities could actually act as a reference for transfer learning and to characterize the heterogeneity of the cells that we bring to the patients. Secondly, detailed gene expression profiling of normal and diseased tissues can explain potential side effects arising due to these cell therapies. So we can, for instance, check gene expression of the targets across the tissues in the Human Cell Atlas and start explaining side effects that we see. Then thirdly, another opportunity, I think, is to use the Human Cell Atlas to identify novel cancer antigens or tumor-specific T-cell receptor sequences, because normally these should be lowly expressed or not present in healthy individuals, but of course be upregulated or clonally amplified in tumors. And then finally and lastly, we spend a lot of time discussing actually a very interesting project, I think, where the Human Cell Atlas could actually collaborate and reach out to clinical and research institutes. They could be involved in harmonizing biobanks and database collections, and for instance, study how characterizing the heterogeneity of the transplanted cells in function of therapy outcome and in function, of course, of the single cell data that are generated could basically contribute to improved patient response. Thanks.
Fantastic. Thank you very much indeed. Great. And now we have the session for the precision oncology, and we have Karuna Ganesh, and is there Sam Pajati online, I presume?
He isn't joining us. But
Okay, fine. Great. Over to you.
this is... Perfect. So we had a fantastic session, very wide-ranging. We're focusing on the fundamental problem that cancer obviously kills a lot of people, and cancer cells and their microenvironments are dynamically evolving over time. And so we're really very much at the very early stage in terms of the application of single-cell approaches, how HCA and efforts like this can contribute to eventual clinical impact in terms of we're just starting to understand the phenotypic landscape of cancer. So that's really where we are at the moment. Most of the studies today are looking at this often at a single time point, and we have a long way to go from understanding what's happening to being able to identify targets and biomarkers to be able to validate these in clinically representative model systems. Do we even have those model systems at the moment, and how good are they? What are they good for? And eventually to drive clinical impact. So we review the state of the field at the moment, as we were talking about. The focus has largely been, most of the studies out there have focused on immune cells for both technical reasons and for clinical reasons in terms of accessibility of blood and other tissues, etc. Metastasis, which is what kills most people with advanced solid cancers, remains largely poorly studied, although efforts are ongoing. Transitions between cell states are not understood, and clinical impact remains unclear at the moment. But the big opportunity here, of course, is to kind of reverse the traditional discovery cycle of going from experimental models and then validating in patients to reversing that and really starting with the heterogeneity of patients as the starting point for understanding what are the commonalities across the heterogeneity that could potentially serve as good targets. Data and tissue access remains a major challenge here because, of course, anything that we need needs to come directly from patients. Getting biopsies from patients is challenging. So how do we get data at scale? And scale is needed to be eventually able to identify rare populations like stem cell populations and resistor populations that might be important to target, despite all of the underlying biological and technical heterogeneity. And of course, heterogeneity, starting with the different genotypes of the tumors, is a starting point for cancer, unlike, for example, in regeneration or development, where things are likely to be more uniform. We also think that embedding within clinical trials is important. Multi-regional sampling is likely to become important. A big question that was raised was one of data quality. Should the field adopt minimal standards for the number of gene reads per cell, for example, the number of cells that should be considered publishable, or how do we interpret these data, or how do we correct quality control and batch process these data? How can we integrate national biobanks, epidemiological studies into our analyses? Many modalities are becoming available. There was a lot of excitement about the move towards being able to do spatial and single-cell approaches from paraffin fixed tissue, which is, of course, what most clinical biobanks have and what most clinical practices do. So that is really going to be transformative for the field going forward, although that also comes with its own technical challenges in terms of how deep the data are and are we capturing all of the relevant data that we care about. And then, as we alluded to, sort of the need for more representative experimental models. Efforts like the one in the Human Cell Atlas to cross-validate across organoids and tissues from, ideally from the same patients, are really, really important and will really be critical to moving the field forwards to know when we start to do perturbation studies, which model systems should be perturbed to actually get meaningful, clinically impactful data. And to integrate the tumor microenvironment and perhaps a systemic microenvironment, which with perhaps transcriptomic analysis may not be good enough. We need to know what are the metabolites the tumor cells are seeing, what are the proteins in the tumor microenvironment and systemically that are changing during tumor progression. How do I change this? How do I move this forward?
Yep.
But thank you. Computationally, there are also major challenges that the field is addressing. Correcting for technical batch it remains challenging, trying to distinguish between biological variability and stress or technical artifacts based on processing remains a challenge. Defining trajectories, these algorithms currently are often validated in development cell state trajectories, which may be more uniform than in many cancers where these trajectories may get perturbed and there may be selections stochastically for specific states that cannot perhaps be easily inferred from single time point data. And so we need more longitudinal data to really try to learn these tumor evolution patterns. And a big challenge is really how do we get from understanding trajectories, from describing cell states, to getting to a small number of testable hypotheses, because ultimately all of these hypotheses that are arising cannot be tested experimentally towards clinical impact, and we need to learn how to prioritize this better. And finally, in terms of impact, perturbation studies and good experimental design to include defined patient cohorts, incorporating single-cell approaches more into clinical trials so that we can understand from the same patient the phenotypic readout of a perturbation, the functional readout of the perturbation in terms of disease control and extension of life. And finally, we are excited by what we've been seeing in the last few years through the efforts of the HCA and others in terms of bringing together people with truly different backgrounds and interests, clinicians, computational biologists, experimental biologists, to really start to move the needle and move the field forward.
Great, thank you very much, Aruna. And so for our fifth session, we have neurological disorders. Is that online again?
Yeah.
So we have Michelle Schwartz to give an update.
No, I was not supposed to give. You asked me to be in the audience, but I can summarize without any slide. So I'll do it with pleasure. So overall, the discussion was very good. There were not many people that attended. I think the focus was whether the mice and human what each can teach us. There was, I think, an agreement that we cannot give up the mice. The mice are very important. In order to be convinced that we can move from mice to human, we need to do multiple models. There was a lot of discussion whether the immune is good and bad, and someone brought the issue of double-edged sword. I think that there was an agreement that we are reaching now a new era where we have to study all sites, all niches of all immune niches in neurodegenerative diseases, including the meninges, the choroid plexus, the CSF, the blood vessels, everything. So we cannot take it as immune system versus the brain, but each compartment may be different. We are aware that doing biopsy in neurodegenerative diseases will be very difficult. So the issue is to be able to focus on immune profile. And we agreed that in many of the neurodegenerative diseases, by the time the disease is becoming symptomatic, in many of the diseases it's too late, such as ALS or Creutzfeldt-Jakob disease, and therefore we need to be able to get access to carriers of the disease, and in order to test their blood for profile prior to the onset, and from this to go forward to sporadic diseases. There was some question about the diversity, the sample size, which is very critical. And I think overall there was excitement around the table that this is because of the awareness of the role of the immune system now in neurodegenerative diseases. There is excitement around the table that we are reaching a new era, that we are far beyond the cancer field, but we are aware that these diseases are not neuronal anymore. They are multiple cellular, both inside the brain, outside the brain, and the brain borders. So that's the major issue, and I think the discussion was very vivid.
Great, fantastic. Thank you very much. Great, so I think we can now open the floor to any questions or discussion, obviously to any of the group that are part of this medical impact session. So please do come up with your questions. Now, while we're waiting for people to put up their hands, maybe I could start one off. I was very fascinated by your suggestion that we can start to predict tumor antigens from the TCR sequences. And I was just wondering, how realistic you felt that was now.
No, I think the way I understood the discussion in the session was that we can use the Human Cell Atlas as a reference, as a healthy reference. Of course, this has to be done in combination with studies in tumors. So I think it was more conceived as a available database where you can then basically look at what happens in healthy tissues.
Right.
So it's not just limited to the Human Cell Atlas, if I...
Dana.
So I'll just, continue with my pet peeve and sort of broken message. But the Human Cell Atlas is a lot more than a big database full of truckloads of data. Along with the data, and we're still lagging behind, is the conceptual and computational methods to properly query it. Because, for example, from the third session, transfer learning and mapping and whatever, it's the conceptual way that you actually model and characterize these cell types. Some of the similarity metrics that Graham was talking about this morning, and some of the conceptual and foundational and technical tools to actually query the atlas that are no less valuable than the data itself, and a big part of this community.
Yeah. We have a question. Yeah.
Hi, this is Zorina Gallus from HubMap. I think one of the things that I'm not sure I saw in the summaries is how important is the ability to pinpoint specific changes in specific cells in the environment, either of the tumor or the cells that are being transplanted or by regenerated cells. So this is something that usually escapes to methods that just make like a big soup of the tissue. So I think that's where a big advance will come, being able to pinpoint which cells are actually responding to the one cell that we think is so important.
Maybe the precision oncology people, would you like to comment?
Yes, absolutely. It is going to be important to distinguish cause and effect, and I think algorithms like the spectra that we heard about from Donna earlier and others to understand how cell states and cell types are changing with regard to each other and communicating with each other will be really important. And from that, of course, to have representative model systems to test those hypotheses with perturbations.
Great. Any other comments or questions? They don't have to be comments. They can questions. They can also be comments on where you see really the medical impact of the Human Cell Atlas first and foremost. Don't be shy.
So this is Aravinda. I'm just. Can you hear me?
Yes, we can.
Yeah. So I want to sort of emphasize this point that Dana just made, that I would think, even though, that what genomics did is really focus the attention of at least everyone, I would say, geneticists particularly, of to go beyond a single gene by single gene effect and to look at the entire genome. I mean, that's what gives us the phenotype. I think Donna's point that we should really conceptually, this should lead to some changes is actually very important. It did come up in our session by a number of individuals that perhaps genetics and cell biology has been apart too long in too many disciplines, and it will force a reckoning that genes don't act alone, but sort of together. And this may really improve the study of very complex processes by reducing, in some sense, the kind of functional universe in which we search for defects or search for meaning. It's not 20,000 independent units, but for lack of a better term, regulatory networks that may be complicated, but are probably far fewer in number. Because anyway, I just wanted to emphasize that conceptual advance would really make a very big difference than currently the way we look at data.
Great. Yeah, thank you for that comment. We had another comment here.
Yeah, so here, Farzin from Hub. For the immuno-oncology field, it seems to me that we are recently coming to the conclusion that only targeting one tumor-specific antigen is not enough. So, for example, checkpoint inhibitor is not very successful. So it seems that now we are looking for a combination of two different tumor-specific antigen. And if within this HCA, we can come up with tumor-specific antigens that are like two of them or three of them, that in combination we can hit them. So bispecific antibodies that from one hand targets drug-resistant pumps, so disable them from one side, and from other side, tumor-specific like checkpoint inhibitors. So if we can come up with a combination of tumor-specific antigens that are both of them and hit them at the same time, then we would be able to have more specificity and less toxicity. So within these two HCA, I was thinking if we are not only looking for one specific, but a combination of tumor-specific antigens for follow-up drug development.
Yes, and of course not just the tumor-specific antigens, but also potentially different cell types that can then come and attack that tumor. Yeah. Do the the oncologists want to comment on that at all?
Yeah, I completely agree. This is going to be very important. I think from a clinical standpoint, we also have to keep in mind toxicity, right, which has been the challenge even with more traditional precision oncology efforts where we think, targeting kinase signaling, for example, with multiple drugs. Yes, absolutely in principle, but it's the practice that's going to matter. And I think there again, to the point we were hearing earlier, perhaps the HCA and others like this can act as references to help us perhaps first and perhaps most importantly minimize toxicity and identify potential on-target off-tumor effects that we might anticipate.
Wait.
We have two specific targets, then we also reduce toxicity.
Yeah. Just to add to that, so one thing that came up in our session was that in addition to just profiling healthy individuals, we would have to profile healthy individuals of different ages with maybe different comorbidities, because there also gene expression can change. And that's, of course, the patients that you will treat. So, yeah, you should see it a bit broader than that, probably.
Coming back to the theme that Barbara mentioned earlier, that data is good, and the more breadth we have in terms of both health and disease and over the lifetime, obviously all adds. Dana.
I guess I'm in a philosophical mood, so I'll continue with my philosophy. I mean, this session was about sort of what we can do, medical impact in the future. And I just want to stress, because it had some highlight here, but not big enough, that, the HCA from the onset has been having medical impact from the beginning. For example, we love to poke at immuno-oncology, so the one sitting next to me already did things from the very beginning. I think the point is what we can do more once we have a fundamental reference. I believe that we cannot really understand disease truly beyond poking and prodding and trial and error without a fundamentally complete and well understood model of healthy. And that does include the comment that was made before, that in order to understand what is aberrant rather than individual variation between individuals that all exist, we really need to have a comprehensive atlas that takes into account the human variation on its different flavors, and then we can properly, with a fundamental knowledge, model, and understanding of healthy, really begin to understand disease in a much deeper and more powerful way.
It actually goes both ways. So often understanding homeostasis is easier when you actually have a disease to refer to.
Oh, disease is a wonderful perturbation.
Yeah, because without the perturbation, it's hard to understand the normal or the homeostatic, I would say. So they kind of go together, especially in human biology, where we don't have access to the types of manipulations that we would do in in the model system. They really go very, very tightly together.
And maybe just to add to that of course for the immune system, the infection and disease to a certain extent is part of the normal behavior of the immune system. So in a way that's not abnormal, but the actual intrinsic function of that system. Any other comments or questions from the audience with respect to possible medical applications or I've got a very quiet audience here this afternoon.
Thank you. Maybe only a comment and extending on what Dana and Aviv said. I think to understand perturbations and this disease and health state, we need it's very boring, but a common framework of annotations that clinicians everywhere in the world, specialists of one disease being oncology or a specific cancer or a chronic inflammatory disease like morbus Crohn, that they agree what are the stages from like 0 being healthy and 1 being normalized as being the terminal end stage of a certain disease, that we can then even start making these connections be genotype or being perturbations or being where these cell types are on this map, on this trajectory of going from healthy, homeostatic to diseased or even aging.
Great, thank you for that. I wondered, I mean, I presume the audience is mostly sort of from a molecular biology background, but we are talking about using the HCA for medical applications. So maybe there's somebody in the audience who might like to comment on how challenging or how practical it would be to actually use single-cell sequencing within clinical trials, and, you know, whether there's anybody who's got experience of that here, and, you know, whether, you know, because that's clearly, in a way, the next step to really take it to the clinic.
Hi, Kirsten. I think it's a very good question. And I can just comment that I think we should include them as soon as we can and collaborate with the clinicians and planning these clinical trials. I think we go back, from my point of view, it's down to the money. Can we get enough money to profile enough patients? I think that will add a lot of cost to the clinical trials, and otherwise I don't see any downside to that.
Yeah. Aviv?
I would just say very broadly that I don't think that this is a money question as the primary one. I think there are many, many challenges before then. For example, we saw it in the summary. They said if it worked on FFPE, everything would be very different, because it has to fit in the context of clinical practice. And FFPEs are clinical practice, not fresh tissue for sure, and also not the type of frozen tissue that we even use for nuclei, and most of the type of frozen tissue that we use for nuclei in most of the spatial assays that we hear about here that are wonderful for most of our studies. So I think first there's some technical limitations that are really a barrier for clinical samples in general. Yeah. Yeah.
I mean,
it depends on
that.
Well, maybe Michaela can respond, and then we'll have Dana.
It definitely depends on the type of organ and disease that you're studying. There will be additional difficulties, practical and logistical, but in terms of the principle of its usefulness, it would be really good. At the same time, I agree, we can't, it's a little bit early to decide to use these technologies to decide if a clinical trials worked or not. It might be, but what it will be definitely immediately useful is understanding how a certain drug or treatment is affecting the patients. So again, it depends how we're going to use it.
So again, you know, I want to again push back on the fact that it's a matter of money, because many of the pharma actually realize this and are actually willing to add a single cell interrogation. The clinical trial is so expensive at the MD and human level that actually the cost of the single cell sequencing is small relative to the cost of the clinical trial per patient, relatively. And, you know, I found companies are actually willing to add that component, the pharma companies, as part of what they do. What Aviv said, you know, access to tissue, FFPE, you know, at MSKCC, you know, in cancer, we cut out a lot of things. There's biopsies, there's surgeries, but still getting quality single-cell data, even when everyone wants to, requires a dedicated and able person that's really dedicated to something. And of course, with FFPE, it would be easier. But really, I think that the problem is, at least for the clinical trials, where the pharma companies are really invested and interested in it, money is not our bottleneck here. It's, you know, logistics and technologies.
Yeah. And again, maybe that's where, you know, again, this community could take a role again in moving technologies to those kind of improvements that just make the logistics a lot easier, you know, starting with frozen tissues, with fixed tissues and so forth. Yes, there's a comment here in the...
Yeah, I just would like to add that we have already included single cell for an actual clinical trial phase 2 authorized by a European medical agency on Asherman syndrome. This is the uterus, and the experience has been outstanding. We have learned far more than we expected because this was proving an advanced cell therapy. So we have scored through single cells the results, and we have really learned a lot. So I think that this is a great idea, and as said, economically wise, is not a huge burden, but what we can get from that, at least for us, has been really important.
That's really encouraging to hear. That's fantastic, because clearly, as a community, we would like to see far more of that. So that, you know, because we all know how much more information we get out than when we do bulk. So, you know, if that gets translated to the clinic, that would be fabulous. I think we are actually coming up to time now. So thank you very much for everybody who's to all the presenters who wrapped up and everybody who's contributed to the discussion. Thank you.
Thank you. Bye-bye.
Yeah.
See you, Daniel.
Okay. Hi, everyone. I'm Sarian Bowers. I'm head of policy at the Sanger Institute. I think probably some of you know me as the GDPR lady, but luckily we're doing science today. So we've got a really great session lined up. We also, remember, we have a lot of participants still online, so I encourage everybody online to be asking questions as well, and I will be checking in, and Sam, remind me if I don't. So our first speaker today is Amanda Oliver, who is going to be talking about the Discover project and towards the human lung cell atlas. Amanda, there you are.
Hi everyone, it's a great honor to be here and representing the Discover Consortium today. And actually this is a slide from Martijn, who sadly can't be here in person because of COVID, but I think I can speak on behalf of him and the rest of the members of this consortium that it's been a really amazing collaboration, generated a lot of data and a lot of insights, and I think a lot of the Discover results have been highlighted already quite a number of times in this meeting, especially, you know, the first draft of the Human Lung Cell Atlas, but also some of the aims and outputs of Discover have been with spatial data, perturbation atlas, using single cell to characterize different in vitro models, and of course expanding upon lung disease atlases. And so I'm going to talk on the spatial side of things today with this really exciting project that I've been involved in the Teichmann lab, and this is really to discover tissue niches in the lungs using spatial multiomics. And so really the key here is that we need to move beyond the suspension cell era to the spatial atlas and truly building a map of the lung and of other organs and tissues. And you know, it's no argument that the suspension cell, single cell, single nuclear RNA sequencing has revealed new cell types and the full transcriptomes of the full complement of cells in the lungs. And you know, this has been exemplified by Fabian, Malta, Lisa, with the integrated atlas of millions of cells, hundreds of donors and almost 50 studies. But what we're really missing here is the spatial context, both at the macroanatomical level and the microanatomical level. And in particular, the lung has quite complex structures within it, and this is important to understand. So the key question that we had here is to understand what are the spatial contexts of the cell types in the lung. And so to answer this question, we profiled the lung in multiple locations across the airways, so trachea to different generations of bronchi, and the upper lower parenchyma. So we're covering that macroanatomical differences. We profiled with single cell and single nuclei from these healthy disease adult organ donors. And we profiled with spatial transcriptomics using Visium across 20 sections from these donors, including 4 FFPE. And this paper is on bioRxiv. I would encourage you all to go and read it, because in 10 minutes I don't have time to really go over everything, and there's a lot of really exciting stuff. We also have lungcellatlas.org to download and browse the data for yourself. And so this is an overview of the suspension cell part of the data, and I really need to acknowledge Elo Matasoon here. She's the co-first author of this work, and she's really been driving the project from the very beginning. And so in total, we had transcriptomes from almost 200,000 cells and nuclei. We identified 79 different cell types and states, and this includes 11 novel cell types and states that weren't annotated or weren't present at all in previous atlases. And this was really highlighted by Malta and Fabian, that when they mapped this data on top of the core reference, there were cells like chondrocytes and erythrocytes that weren't present at all in any other studies. It's not that they weren't annotated, they just weren't captured. And this is a strength of our sampling method, including things like single nuclear RNA sequencing. And not only are there novel cell types and states, we also have enhanced resolution of particularly immune compartments. And again, this is the complementarity between a focused study like this and a large integrated study, where in the integrated atlas, you recover more rare cell types, but in a focused study, you can have this finer granularity, and both are required, I think. And so we have this really nice fine-grained annotations across the lung in these different locations. Now, putting the cells in their place and discovering these spatial microenvironments. In this study, we use Visium, and this is an example of a Visium slide from the trachea, where you can see the H&E, that on top of that, we have the Visium spots. And really, with this data, we can do manual structural annotation to look at the microanatomical tissue structures and features like epithelium, glands, cartilage, muscle, things like this. And then we can perform unbiased analysis using cell-to location, where we can take our suspension cell data and map these cell types and states onto the tissue context. And using these two modalities together, we can then co-localize cell types and states into this tissue context. And so first, we were really able to show that we could identify known cell types in their expected locations. And I think this is really important as a kind of proof of concept that these techniques are really working quite nicely, and the algorithms and the computation is working. And so here, this is a bronchi section. We have, with the pink arrow in the center of the H&E, is the epithelium. On the left-hand side with the green arrow, we have the parenchyma, and then these brown arrows are pointing to the cartilage. And so then with the cell-to location analysis, you can see that ciliated basal cells, they're mapping to the epithelium, AT1 and AT2 cells to the parenchyma, and then chondrocytes, which were a novel cell type in our data, these are the cartilage-producing cells, and they're present within the cartilage in the airways. And so this is an example of one section, but we can also look across all of our 20 sections and look at the cell type abundances in these manually annotated regions to again show that, AT1, AT2 cells are mapping exclusively to the parenchyma. And so the confidence that we have in these technologies means that we have the scope to discover novel cellular organization. And a really key novel aspect that we found in our study is this gland-associated immune niche, or GAIN. And this is really central to the submucosal glands, and this is an example of, or a schematic of the structure of these glands that produce mucus in the airways. And there's a number of epithelial cell types that are involved in this gland structure, and these are duct mucus serous and myoepithelial cells. And in particular, the duct cells were, again, a novel transcriptome that was annotated in our data, but not in other lung atlas papers. And so really excitingly, we identified that IgA plasma cells were mapping exclusively to the glands, but not other tissues. And IgG plasma cells, for example, were not present in these gland areas. And so this was really exciting because it wasn't really known, and these IgA plasma cells are co-localizing with the submucosal gland epithelial cells. And so using this spatial data with the full transcriptomic data, we're then able to kind of interrogate the signaling mechanisms that are involved in this niche. We can do this with cell-cell interaction analysis from the full the suspension cell data, and then this can drive the hypotheses and validations of the signaling circuits involved in this niche. And so I don't really have time to go over all of the kind of validation and exciting data and nuances of this niche, but again, I encourage you to go to our paper, it's on bioRxiv. But as a schematic representation of the gland microenvironment, again, we have, again, this submucosal gland structure which produces mucus to line the airway surface, and the various epithelial cell types involved. And these epithelial cells express chemokines that are key for recruiting immune cells to this niche. And then when these immune cells are there, they're maintained by key signaling molecules IL-6 and APRIL, which are important for immune cell survival and IgA plasma secretion. And so we also found that these epithelial cells can actually present antigen CD4 T cells via MHC class II, and the culmination of all of this immune activity by these orchestrated by these epithelial cells is really increased IgA production at this site of mucus production, which makes a lot of sense. You know, we want the antibodies to be in the mucus. Why don't we make this all happen at the mucus-producing glands? And overall, this is really to protect from infection. And we know that IgA is protective for viral infections. It's also important for vaccine responses. And this niche is also likely to be implicated in diseases like cystic fibrosis and COPD. And so taking a step back, I really wanted to highlight the power of spatial atlasing, where we had our really nicely annotated cell types and states from the suspension data. We then mapped them onto the tissue context with spatial transcriptomics, and then actually that feeds in to enhancing our resolution of our cell type annotations. And this is one particular example where we had fibroblast populations that we had annotated. We could see they were separate clusters, they had specific marker genes. When we mapped them onto the spatial context, we could see that they were in really specific locations, like around the bronchi or surrounding the cartilage. And so then we call them peribronchial or perichondrial fibroblasts. If not for this spatial information, maybe they're fibroblast 1 and fibroblast 2. And so this really highlights that you can identify new cell types and enhance the resolution of your annotations. Identify novel tissue niches like the gain, and something I didn't really touch on but I think is also really important is identifying spatial gradients related to organ function, such as immune response and oxygenation for the lung, for example. And again, I'd just like to highlight that you can download and browse all of this data yourself at lungcellatlas.org. And lastly, I'd really like to acknowledge this was a huge team effort. I already gave a big shout out to Elo. I can't acknowledge her enough. My partner in this project, of course, Sarah and Kirsten, the whole team, funding bodies, Discover. Again, it's been a really amazing collaboration, and will continue to foster collaborations going forward. And of course, to the donors and their family for the tissue that we analyze. And yeah, happy to answer questions and have further discussions in coffee.
Thank you so much. As that was an incredibly timely talk, we have time for questions. Does anybody have any questions? I see, do we have one online?
It's not so much a question as a comment from Giorgio Cataretti from the University of Milan. Trachea is a known target for extramedullary myeloma plasma cell tumor.
Okay. So yeah, I guess that's something. So one of the things that we haven't done yet, and is kind of future directions, is to really interrogate this niche and other novel tissue niches in disease. And so this is one example of where we can try to see, this is what's happening in the healthy situation. How does this change in disease? And then maybe how can we target that? Going back to the clinical applications of these atlasing studies.
Great.
Thank you for that was a really great comment. Thank you.
We have a question over here.
So I'm really interested, how can you squeeze so much information from Visium data whose resolution is not that good? So I don't know if you had some comments on that. Definitely the resolution could be better, but I'm really glad to see how much you could get out of that. So with the right tools, you can do a lot of things, or there are some limitations that you still, this would have been better?
So, I mean, yeah, I think we do sometimes get comments of, you know, there's Visium HD that's coming out and that's going to surpass these technologies, but we're showing as a really clear example that you can find interesting, novel biological insights with these 50 micron resolution. And I think, you know, there's limitations to any technique. We've found that, for example, I think plasma cells map really well because some of the marker genes are IgA. It's constantly secreted, so the transcript is high. Same thing with, you know, other secretory cells like the mucus cells of the gland. Sometimes we miss T cells and things like that. So for example, I showed in my schematic that we know T cells are at the gland. We couldn't identify that with Visium, but then we can, you know, we see the IgA plasma cells, maybe T cells are involved, and then we go to more traditional IHC, RNA scope, single cell resolution, what we can do.
Any other questions? Oh, I think we have one online again.
Thanks. This is from Heather Etchevers at INSERM. How do you see the next steps in integrating this version of the lung cell atlas with the developmental atlas?
That's a really great question, and I think I'm also going to throw that to the wider audience to think about as well. Yeah, I guess I don't have a good answer for that question. I haven't thought about that in much detail, but I guess a key thing is that we need more data to integrate and see how the spatial differences and, you know, I guess developing lung, for example, has its challenges because, you know, and this is also on the resolution of Visium, 50 microns worked out well for the lung. For other tissues, maybe not. And for developmental tissues, no, because everything's smaller, so 50 microns proportionally is maybe not going to show as much.
Great. Any other questions? Okay, if not, can we have another round of applause for Amanda and her wonderful talk? Good job, well done. Oh, next up, if I can find my emails, we have Rosa Vento from the Wellcome Sanger Institute, who is going to be talking about the single cell roadmap of human gonadal development. I believe she's online. And we'll be giving her talk virtually.
Hello everyone, it's a pleasure to be here and present the work we have been doing as part of HugoDec. So to begin with, what are the gonads and where are they coming from? So the gonads are quite a unique tissue because they face a developmental choice. They need to decide if they become a testis in males or an ovary in females. And as you can appreciate here, they have different morphological and even different functions. So they serve both as a reproductive tissue by being the microenvironment where the gametes, which are the cells that will carry the genetic information to the offspring, resides, and also as an endocrine organs by releasing the sex hormones. So up to 6 post-conceptional weeks, the gonad appear as undifferentiated, and depending on the genetic content, they will give rise to the testes or the female or the ovaries. So then the gonads are a fantastic organ to study this bidirectional interplay between the cell identity and the tissue microenvironment. So the sex determination starts in the somatic compartment of the gonads around 6 post-conceptional weeks by the activity of SRY that causes the differentiation to the male Sertoli cells. On the other hand, if the individuals lack expression of SRY, then the default female program is activated, and this leads to the female granulosa cells. So what is key is that the sex determination starts in the somatic lineage, and that will instruct the germ cells, the precursors of the gamete, to either go into pre-spermatogonia in males which will assemble with Sertoli cells to form the cords, or with the oocytes which assemble with the granulosa cells to form the follicles. And there are multiple differences on how about the process in male and female, and one that I want to highlight is that in female you have this mitosis to meiosis transition, meaning that in females we are born with a specific number of gametes. So the majority of work so far has been focused on the germ cells, and there is little known about the somatic lineage. And it's very little known also how this compares to mouse and how we can translate findings between species. It's also very little known how these somatic cells talk with the germ cells. For example, how they do this in the follicle, and this is relevant for knowing the conditions where these germ cells differentiate and important for the development of in vitro gametogenesis models. Finally, it's very little known about the immune cells, and this is something we have looked in our work, but I won't be presenting today. So we have made an effort to do a comprehensive map, collecting samples from both human and mouse that cover both the sex determination stage and the parallel differentiation. And we have profiled more than 50 donors in humans and using a multi-omic approach, which includes RNA attack and also spatial data. And we aimed for a harmonized annotation across the species, which means that we annotate the same cell stage in both the species, and there were a lot of differences between the markers, and that's why to do that we also used the temporal dynamics and also went back and forth with the spatial data. And thanks to this harmonized annotation, what we can do is to compare the gene expression between species. And then what we did was to train a machine learning classifier on the human data and project it into the mouse. And this allowed us to compare how similar the populations were. And what we observe is that the somatic compartment, and in particular the early somatic compartment, is quite transcriptionally divergent between species, and we examine this further. So then we zoom into the somatic population in both species, and we find new populations actually in both species. And I want to highlight one of them, which is this purple population, which you can see is transient, that appears both in male, females, and both species. So to know more about this, what we did is to use real time and also pseudo-temporal time to see where it came. And with this, we made a model by which the coelomic epithelium in the surface loses its mesothelial features and acquires somatic features. And these early somatic cells give rise to the purple populations, which will further give rise to the Sertoli cells in males or the granulosa cells in females. And something that supports this theory is that this population, these early progenitors, are the first ones that in males express SFY. And we see markers different between species, quite a lot. And that is relevant because some of the markers are actually genes that are involved in differences in sex development, highlighting the translational impact of our work. I won't go into detail, but also we define this early supporting PAX8 lineage, which will derive into the rotodestis in males, which is this population between the gonadal and extragonadal tissue. So let's move to the second question, which is about the crosstalk between the germ cells and the somatic cells in the forming follicle. And here again, we zoom into the granulosa lineage and we define novel populations. And to compare them with the other species here, we looked further into the transcription factors, and we look at their expression and their activity by making use of the multi-omics data. And again, what we could see is that there are specific regulatory programs actually in primates that are not conserved in mouse, just highlighting how relevant is to study this process in humans. And as happens for a lot of different developmental processes here, we also have two waves of differentiation. And we managed to do that by looking at the spatial data. And what we could see is that the first wave of differentiation in blue is restricted to the medulla. And this one will originate from this progenitor that I discussed before. But also in the second trimester, we also, or later the first trimester, we have a second wave that comes from the cortex of the ovary. And they start differentiating into more mature cells as they go into the ovary. So granulosa cells compartmentalize the ovaries, and actually this goes hand in hand with a compartmentalization of the germ cells that we and others defined. And what we then want to understand is how this communication, this dynamic communication happens. And what we did is to use self-NDB, our method to look for ligand receptors interactions, but we updated this method by also including transcription factors, and we used these as sensors for the activity of the receptor. So we restricted the communication to cells types that are coexisting in space and time. And what we did is to define 3 microenvironments and the interactions that are happening in each microenvironment. And here we can see the different ligand receptors represented by dots. And what I want you to gain from this is that what we see is that in the outer cortex, we have an environment where the immature germ cells can survive. But then as we go into the cortex, we have molecules that are relevant for this meiosis and this formation of the follicle finally, and the survival of cells there. And this is also super exciting because then we can use all these receptor ligand interaction, these transcription factors, and also the ECM as a recipe to develop new protocols for in vitro germ cell maturation. So as a conclusion, so what we have done is to align single cell atlas of developing gonads in human and mouse, and this has allowed for a cross-species comparison. And thanks to this quite accurate temporal resolution and also to the multi-omics data, we have managed to see that the developmental processes happening in mouse and humans are quite similar, and we define new cell states. But actually we see that the regulatory programs that will mediate these cell states in mouse and humans are quite different. And again, I didn't go into details. We also find novel immune cells, and we are excited for all the translational impact for the in vitro gametogenesis, for the translation of the mouse and humans, and also for gonadal conditions such as differences in sex development. And just to finalize, a big thanks to the dream team, Valentina Lorencia and Luz Garcia Alonso, who led this work, and the lovely images from Cecilia Coresi-Maceo, and also to all collaborators and the Higodeca Consortia. Thank you.
Okay, do we have any questions for Rosa? I see one over here from Jonah.
Jonah Cool, CZI. Great talk, Rosa. I'm curious if you have any sense of what those PAX8 progenitor cells become or contribute to, and then maybe the spatial temporal regulation of them or those specification and fate decisions.
Yeah, thank you very much for that question. And actually, that's an example where spatial was key for us to understand what this population was doing. So by looking at the single cell data, we saw that they have a lot of specific receptors and especially ligands, a lot of secretory molecules. And then when we look back at the tissue, they are really interface between the gonad and the mesonephro. And so it changed a bit between the first trimester and the second one. So in the first is common in both male and females, and we think that they communicate a lot with endothelial cells, and they may be kind of a gatekeepers of what comes into the gonad. And then in the second trimester, they disappear in female, and then they are in male, just kind of sucking the testes. So in a way, we think it's where the red testes are located. So it's kind of the population that will help with all these red testes formation. And you can see these are lovely in the 2D and 3D images. And this also makes sense because it's described that in female, you have an early red ovary that then disappears. And yeah, so basically the spatial was very key to kind of assume the function for this. Thank you.
Thank you. Any other questions? Oh, I see one over here.
Hi, Hari Nakshatri from Indiana University. Do you see a specific pattern of the keratin expression, in the sense in the breast we start to see the basal luminal hybrid phenotype where different types of keratins are overexpressed in a particular cell type? Do you see a similar pattern here?
So I'm afraid we didn't look at the keratin, but we can easily look. I also, like, all the data is available in reproductivecellatlas.org, and you can query all these data sets. So I'm happy to look at this, and all of you are free to look at your gene of interest. But thank you for the comment.
Any other questions? I don't think we've got anything online. Okay, if not, I'm going to, in the interest of time, I'm going to move us on. Can we all thank Rosa again for that really interesting talk? Our next talk is from, and I apologize if I get your surname wrong, Barbara Trutlein. Good enough. And she's going to talk to us about her results from the brain time work, a multi-omic atlas of human brain development.
Thank you very much. Yes, it's my great pleasure to present on behalf of the Brain Time team. And you can see here all the labs that are involved in the Brain Time consortium. It's Stan Linnelson's group from Karolinska. Then we have from the Weizmann, Ido Ahmed, Amos Tanay, and Udi Shapiro. And Ed Lean from the Allen Institute for Brain Science, and Cartana as a company that has been involved. And so the overall goal of the Brain Time Consortium was to establish a comprehensive spatial and multi-omic and temporal map of human brain, of the human brain from development to aging. And we had a particular focus on the monoaminergic system, which spans the midbrain and the brain span. And so the samples that we have been working with in the different labs span from fetal brain through adult brain to aging brain of humans, including some neurodegenerative disease brains, but also organoids, brain organoids. And then, of course, we have a various set of technology platforms that we have been using and also further developing, as well as analytical platforms, ranging from multi-omics, spatial omics, cell-cell interaction sequencing, perturbations, and lineage tracing. And so I want to highlight today the work that we have been doing in my lab and in Stan Linnelson, Stan's lab, that focuses on the development. And so I want to start by actually showing a few slides from Stan's lab. So he has been pushing the generation of atlases of primary human brain development, both in the transcriptome, but also in the accessible chromatin modality. And so they have been generating these two very large comprehensive atlases. And in particular, I want to point out Camille Mannens from his group. And so this spans the time points from 4 to 13 weeks post-conception and incorporates all regions of the human brain. And this is all still unpublished, so, and analysis is still ongoing, but I just want to show a little bit. So here, just showing quickly the pipeline now focusing on the attack atlas. Of course, after quality control of the data, they have been binning the genomic data and then performing pre-clustering and peak calling, and then reducing the dimensionality and clustering and annotating the clusters by doing actually gene expression imputation. And then they do downstream analysis, and of course here they go in iterations to further zoom into individual larger clusters to identify subpopulations. And well, then what you can see here is then a set of, different gene activities here integrating over all the attack signals in nearby a gene. And you can appreciate that there are inhibitory and excitatory neurons present. We have endothelial cells, we have immune cells, we have oligodendrocytes and astrocytes and so on, all the cell types you would expect. And what was another goal was actually to identify cell type specific enhancers that actually could be potentially even used in the clinic to target in gene therapy specific cell types. And so here just showing 4 examples of such cell type specific enhancers for Purkinje neurons, hindbrain excitatory neurons, OPCs, and floor plate. And currently validation is being performed in tissue explants. And then finally, of course, one can now look into gene regulatory interactions, and what they found was a set of around 10,000 candidate co-accessibility networks that were rather local and regulating single genes. Here showing, for example, tyrosine hydroxylase that is expressed in dopaminergic neurons and its interactions with putative enhancers and tumor genes. And so this is great. But what we can't do with this atlas of primary tissue is, of course, go in and now perturb it and really understand what do individual regulatory regions do, what do individual genes do, what is really the functional importance and role of these. And so this is why in BrainTime we have this organoid system incorporated as well, where actually with organoids, we discussed this already a lot, we can actually, when we grow them from these pluripotent stem cells, we can model development in the dish and thereby learn about human development. And in this case, actually, we grow brain organoids with an unguided approach, where we showed in the past that really from pluripotency we can then generate various different brain regions composing different cell types, such as cortical excitatory neurons, ganglionic eminence-derived inhibitory neurons, and diencephalic mid and hindbrain neurons. And so this was all done on transcriptomic level so far, and so for BrainTime what we wanted to do is complement that now with also epigenomic data. And so we, just to mention, I think we discussed this, but the great thing about organoids is that we can then actually perturb them. We can access time points that we might not have available from primary tissues, and we can perform perturbations, model disease, trace lineages, and so on. And so we decided to generate a new data set here that would incorporate both modalities, transcriptome and accessible chromatin modalities. And you can see now directly the embedding here. And again, we see now very early brain development, really the establishment of neuroectoderm, and then the neuroepithelium emerges, it gets patterned, and we see different brain regions and brain regional identities emerge. And so this is complementing Stan's atlas by adding actually earlier time points. And now we can actually use, for example, cell rank to really identify the branch point. When do cells become restricted for a given cellular identity? And we can see how the neuroepithelium gets patterned into non-telencephalon, telencephalon, and how the telencephalon further gets patterned into dorsal and ventral telencephalon. And then, of course, we can now assess in these different branches all the genes that are expressed, all the, how certain motifs are, transcription factor binding motifs are accessible, and so on. And So actually, I should mention that in this unguided approach, we see a lot of telencephalon represented, dorsal and ventral telencephalon. We have not so much of the non-telencephalic regions. So we wanted to complement this data set with guided organoids, especially because we wanted to look at more midbrain region and brainstem-derived cell types. So actually, we generated another multi-omic atlas. So here, again, measuring RNA and attack from the same cells in midbrain organoids. So all we do, we follow a very similar approach, but we provide the morphogens to pattern this into a midbrain region. And we actually have two protocols. One makes more dorsal and one more ventral midbrain. And so together, we can actually have then a really nice representation of the developing human midbrain. And again, also have cell type specific enhancers present. And on the example of this midbrain data, I want to show how we can bring the primary and the organoid atlas together. And this is really ongoing work. This is still preliminary, but we can use the annotations from Stan's primary atlas and project them to our organoid atlas, and then also assess the confidence of the projection. So are there situations where the projection fails because maybe we have a cell type in vitro that's not there in vivo? And so this is what you can see here now, the projected cell annotation from primary to the organoid atlas. And I mean, this might not tell you much, but what you can see in this heat map now is that we have the primary annotated midbrain cell types and the organoid annotated midbrain cell types. And we see actually great correspondence in all the neurons and the neuronal the neuroprogenitals actually. And here this shows how similar, how good the projection actually is. We have a bit less confident projection to these non-neural cells, such as mesenchymal cells in the organoid and neural crest derivatives, which very much makes sense. So overall, we can actually say that we find a counterpart for every in vitro cell type. We find a counterpart from the in vivo data, which is very, very nice. And also, when we look into the accessible chromatin data, actually looking at peaks that we annotated completely independently, actually, and again, still this is preliminary, but if you look at this, we actually find the same peaks in the primary data set, in the organoid data set, and it will be fun to really quantify similarities, as we discussed earlier, that we need, in that case, additional tools potentially. And so then just in the last few slides, what can we do now that we have organoid data that is actually recapitulate aspects of primary brain development? We can do manipulations. And in this case, we actually started by developing a tool to infer gene regulatory networks, which we call PANDO, which really uses both modalities together to model the relationship between transcription factor expression, target gene expression, and binding site accessibility. And using PANDO, we can then infer a gene regulatory network for our early organoid brain development data set. And here now just showing transcription factors, but of course we can now look into each transcription factor, what is the module it is predicted to control, and what other factors does it interact with. And because we can perturb the organoid, we wanted to test the predictability of this network. So what we did is we used a mosaic perturbation approach, crop-seq, in these organoids to knock out, in this case, 20 transcription factors that have an interesting expression pattern across cortex development. And when we look at the data, where we again have the different branches of cortex, ganglionic eminence, and non-telencephalic cells, we can see that the perturbations lead to enrichment and depletion effects for these different trajectories. And so we have some genes that, for example, GLI3, that really, when knocked out, leads to a depletion of the cortex. And a gene such as HAS1 leads to an enrichment of the cortex and depletion of the ventral telencephalon. And here you see the guide detection. And so this is actually very interesting, and we wanted to go back to the GRN to now ask, could we have predicted this? And indeed, when we look at the gene regulatory networks at the decision point where telencephalic cells get restricted to either dorsal or ventral lineage, we can see that GLI3 in that state activates a lot of genes that control cortex development. And when you knock it out, you don't get cortex, which is what you would expect as predicted by the GRN. And HES1 actually has repressive interactions with cortex genes correlating with the cortex. And so overall, we could actually dive in even more and really look at the genes that are perturbed in the GLI3 knockout case. And these were all targets that were predicted from the GRN as direct or second layer interacting genes, which was very encouraging that actually these GRNs can be meaningful. And so this I will skip over. It just shows that we develop tools to do lineage tracing, which we can do in the organoids and not so well in a primary human tissue. And so with this, I'm at the end, and I thank the whole consortium, and I'm happy to take questions.
Thank you so much. We have time for one question. I think Muzz has nabbed it.
Thanks, Barbara. Great talk. I was wondering, I sneak in two. The first is, have you correlated the organoid weeks with the developmental weeks in terms of what are the corresponding timelines? And secondly, you don't have microglia in your organoid, and I wondered what you thought microglia were.
I thought even to add a microglia slide, but I thought I will go way over time. Anyway, so currently we don't have, in this data, we don't have microglia present. Actually, in this consortium with Ido Amit, we have been actually looking at the primary microglia and then also differentiating microglia in vitro and putting them into the organoids and assessing similarities and how this brain microenvironment changes the state of a microglia. So yeah, that's definitely an interesting aspect. And the timing, well, actually we compared similarity between we assessed the similarity, and we found actually in our midbrain organoid data, towards the end of it, the similarity drops down a bit. And then we realized that we are actually, we went up to beyond 6 months of organoid development, and Sten didn't have such late time points in his data. So actually, we think this drop in similarity means that, well, we don't have the matching primary time point, and I think we have quite good correspondence, but I didn't yet fully analyze the clear time point similarities. We should do that.
Wonderful. Okay, in the interest of time, I'm going to move us on, but thank you again for that wonderful talk.
Thank you.
So, sorry, I've lost my speaker. The next speaker we have is Carlos Simon, who's going to talk to us about the Human Uterus Cell Atlas project.
Thank you. Thank you. It's a pleasure to be here. Thank you for having me. I'm going to present the Human Uterus Cell Atlas on behalf of the whole consortium to all of you. This is the profile of this European Union project that unfortunately ends tomorrow, funding-wise, I mean. So we have been 6 partners from countries, and this is just the profiling and the objectives of the objective of this consortium was really to create this single-cell multiomics atlas of the human uterus in health and disease. This is the consortium itself, composed by these 6 groups from Estonia, Andres Salumets from Uppsala University, Cecilia Lindskog, University of East Anglia, David Monk, Roser Bento from Sanger Institute, Sergio from Bahia, and from Incliva, from Valencia, Spain, myself on behalf of all of them. We focus on this organ. This is the human uterus. This is a non-vital organ, but indeed is vital for the continuation of the species, and in a more proxy way is vital for the baby to be born. So this is a really a highly regenerative organ that allows that women will have pregnancies even in the 80s. It has been already proved that. And this is because there is a mucose, which is the endometrium, that his main aim is just to prepare the window of implantation for the embryo to attach, and that's why it's so challenging to understand the niche and how it works. And this myometrial layer, which is the myometrium, that allows the organ to go from 7 centimeters to more than 50 centimeters when pregnancy occurs. So this is just a really enigmatic organ. So the whole consortium, this is just to show you that the tissue procurement has been obtained in 100% of the cases. We have even obtained whole uterus samples from deceased patients, and this is because we have an excellent network of transplantation in Spain. We produce more organs for transplantation in Spain than European Union and USA together. So this allows us to have the possibility to go for that. And this is just the way that it works. Altogether, we have obtained and analyzed more than 6,000 cells, in addition to another that comes from the Sanger from Bento, only on endometriosis. So this is the cells that we have already worked with. And we have been working, as said, in health and disease. For health, one of the projects has been to identify the progenitor cells that they allow for women to menstruate every month during their whole life, including the postmenopause, if we give a hormonal replacement therapy, just breaking 2/3 of this compartment and going back again. So this is a really amazing project trying to find those cells, and by using this trajectory inference with RNA velocity and cell rank, we have been able to pinpoint a specific profiling of cells that we try to understand further using organoid production, and then, of course, single cell from the organoids to see at what extent we can really create this whole endometrium from these specific cell types. As a pathological situation, we decide 2 of them. One in obstetrics, which is preeclampsia, which is a disease that affects in the 2nd or 3rd trimester of pregnancy. Blood pressure is elevated, and patients, we have to stop pregnancy by a C-section, regardless of the time of the pregnancy itself. And as you see here, there is the hypothesis that is the soil, the decidua, the endometrium, that there is a defective decidualization, and there are proof of concept about that it happens. So we really want to learn about that, and that's why we have chosen this disease. These are the patients already analyze all of this sequence, and we are analyzing the data nowadays, but with the partial analysis of data, preliminary results, we are able to really identify specific cell types with, they have even with the same cell types a differential expression. So we are trying to really understand the difference between this defective stage and at what extent, if we have targets, we will be able to really modify the situation. For a gynecological condition, and this comes from Roser Lab, they are working on endometriosis. So they have been able to get this biobank of cells, and they are just getting those cells analyzed from cases and controls, again trying to find out, according to the different stage of endometriosis. Endometriosis is a disease that affects 35% of patients, including women in this audience, and we do not know how it works. But endometrium gets out of the endometrium of the uterus, and it produces infertility, pain, and bleeding. So I think that this is key, and they are working on this set for this specific pathology, and also adding the spatial transcriptomics to the whole consortium, which is very, very helpful to really locate at what extent those differential cells will be regulated. Immune system, it is getting really important in understanding this disease, so that's why it's also amazing the work that could be done in this. And the myometrium, we have been already working on that. As you see, this is mainly from the whole uterus, from diseased patients. We are able to do the cartography of the uterus, of the myometrium, posterior, anterior, and fundus. And we have already analyzed those cells, and we have been using not only single cell, but also nuclei, and being able to start to understand the different cell types that we have in the myometrium in the different geographical locations, and at what extent this could make us to understand what myomas, which is a tumor that 70% of women, they have, benign tumors, may occur. So we are working on that too. And our partner from Uppsala, she's trying to get from those targets from RNA-seq, she has been trying to acquire the proteomics from that, and she has produced 2 different of 6-plex antibodies panels that they will be able to differentiate both epithelial and stroma, and she's working on that. And also incorporated the imaging that comes from those panels to maybe be able to locate at the protein level what we are just trying to have as a spatial transcriptomics and also at the single cell. And our partner, David Monk, is giving us the methylome, the epigenetics of all of these conditions. And what he has done is trying to improve by single cell methylation profiling at single cell resolution. And he has been working on that. And now she's ready to go for that. And these are some of the data that he's collecting in order to go as soon as possible to the cells that we have already collected. They are in the process of being analyzed. Finally, our partner at Bahia, who is a company, a private company, has produced our human cell data portal, in which we have incorporated all the data that I am mentioning to you from all the partners, and we are trying to analyze all of them, getting together on that. Unfortunately, the funding for this will be ended at the moment that the funding of the platform it's finished. We try to bring this data to the Human Cell Atlas, but unfortunately it was not possible because those patients, all of them, should have diseased patients. So we are trying to understand how could be the best way to transfer all of this data to the Human Cell Atlas, other than the general repositories that we maybe use when we publish our results. And of course, we have done some of the social media and also general getting in touch with the public, explaining our project, explaining why the uterus is important and why single cell can give us a different advantage, a new view of health and disease in this organ. So with that, I just want to thank you for your attention, and this is just the faces of the team that they have been working for the last 2.5 years on this project. Thank you very much.
Thank you. A wonderful project on a very under-researched area. Does anybody have any questions? Any online? Gosh, I think we're beginning to flag a little bit at the end of the day, but really, not a single question? Wow. Okay. Well, thank you so much. Can we give you another round of applause? Our next 2 speakers are Timo Trezer and Diego Balboa Alonso, and they're going to be talking about the SPACE project, or SPACE project. I do apologize.
SPACE, I hope. There's a lot of discussion.
Yep, and I will hand over to you. Thank you.
Okay. Hello, everybody. Yeah, we are very happy to be here and show you some of the efforts of the European Pancreas project SPACE, and some of the updates and where we're standing at the moment. Just a brief recapitulation of the pancreas. I mean, it consists basically of 2 large compartments. It's the endocrine compartment, which is about 5% producing all the hormones, and the really large 95% of exocrine compartment, which gives researchers all the headaches, because basically the tissue starts degrading as soon as you work with it, because of all the digestive enzymes that you have in there. So we kind of would like to do the acknowledgements up front, because of course there's not only the work of us 2 or even a few people, but really a large consortium all over Europe. And we couldn't list all of the people, but here at least are the lab heads, who all contributed to the main objectives that we really had in this consortium, which is, for example, the sample procurement, which is especially difficult for pancreas, and manage that, and then really profiling the molecular cells, and then also translating this to cell maturation, development, and also going to the disease states with type 2 diabetes. So we basically had three main groups of patients. So the biggest group, the adult organ donors, which are healthy pancreas donors, where we did single nucleus sequencing. Then there's the fetal group, which have developmental ages from about 13 to 17 weeks after gestation. And then the disease group with healthy and diseased type 2 diabetes patients. And from the adult group, we really tried to capture the whole pancreas. So we took samples from all over the pancreas, so from 4 main regions. They had the tail, the body, and the PP-rich region. From the fetus, since there's so little material, we really had to take all of it. And on top, especially for the disease patients, we also took islets out of the fresh tissue and then sequenced these. So using special methodologies to really isolate the single nucleus out of this highly RNase-rich tissue, we could basically build our reference, which nicely recapitulates the 90 to 95% of exocrine compartment, 5% endocrine compartment, and the rest other cell types. So, and then we applied kind of what most people did here, a hierarchical approach to cell annotation. And I only show you the first 2 levels here, and afterwards it gets a bit fuzzy, as we always heard over this meeting as well, and this is kind of work in progress. We nevertheless used this data then, so the transcriptome of the single nuclei, to also inform basically the other modalities that we had in this consortium. So we also took single nuclear attack sequencing from the adult pancreas, and then it was kind of back and forth, informing the attack sequencing from the RNA sequencing and so on to define the cell types, and then also now defining cell states. Going further into the isolated adult islets, we really again use this basically to inform now the disease states, and had done a single cell RNA sequencing of whole cells this time, not only nuclei, and single cell attack sequencing, and now venturing into comparing this to our reference. And with this, I hand over to Diego. He will give you more insights into the questions.
So one of our interests is to understand a bit better what are the heterogeneity in the endocrine pancreas subcompartment, and for that purpose we have also performed BASA-seq on a subset of human islet samples. And this technology is profiling full-length messenger RNA, enables us to have a bigger depth and capture per cell, and also to study splicing. This is offering us a high resolution to annotate better the cell states, as you can see here, for the beta cells. This is particularly important in the context of disease and understanding the function of the cells, which in this case is critical for diabetes. We also have some preliminary results on the fetal pancreas dataset. This is about 100,000 cells that we have obtained through different stages. And it's really interesting to see that the time resolution of this dataset is already enabling us to see interesting developmental and interesting biological features, like the differentiation and development of the pancreatic progenitors into the pre-acinar and acinar compartment, but also into the endocrine lineage. This is one of the longest-standing questions of endocrine pancreatic development, how the different lineages in the human islets get allocated and specified. And we think that querying this data set, which will be integrated with single cell attack, will inform us how actually these decisions are happening. It's interesting to see that we are able to capture the master regulator, a transcription factor that is transiently expressed during endocrine development called neurogenin 3, and how the expression of this transcription factor is followed by the allocation to the different lineages, beta, alpha, and delta cells. We are really excited about this, and we are just starting to explore ways of integrating this with other bigger data sets, which will inform not only the development, but also, for example, the generation of stem cell-derived islets. Another interesting message that we have found through the study of these single-cell data sets is that it is likely that there is no such a thing as an adult human pancreas. And the thing is that when we have profiled human healthy pancreas, adult, about half of the pancreas that have been analyzed have been assessed by expert pathologists, and a lot of different micropathologies have been found in these samples. So even though that microscopically and there is no history of pancreatic disease in these donors, we can find that about half of them have some kind of lipomatosis, which is the infiltration of cells that accumulate lipids, some kind of fibrosis, maybe small lesions that are a scar tissue of a previous insult to the pancreas, or small pancreatic intraepithelial neoplastic lesions. And we think that now this offers the possibility to capture these maybe early disease states, and for example, we can see that already at the single cell level, that in some of the samples that have been classified for fibrosis, we see an expansion of the fibrotic cell type compartment, and we are now trying to study what are the transcriptional changes that are behind this, and how we can leverage this to inform the decisions at the clinic. The other thing that we have is this multiplex immunohistochemistry using tissue microarrays, where a set of panels of antibodies from the Human Protein Atlas has been validated and tested, and at the end we have been able to come up with a 25-plex antibody panel for this codex, and we are now using this tool also for discovery of and interpretation of the different tissue samples that we have collected, and also for validation. So overall, now we have collected within this SPAS consortium close to 1 million different individual cells. Our interest is to perform an in-depth characterization of the different cell types and states in the human adult pancreas, and by integrating the different experimental approaches. Of course, this imposes a challenge, and that we are working on it to integrate data from the single nuke, the single cell attack, and also from VASA-seq and other spatial information. At the same time, we have the pancreatic fetal development data set, and both at the single cell RNA sequence and single cell attack level. And I think that this is offering us a new window to understand human pancreatic development. The other aim of the consortium is to identify the transcriptomic and regulatory changes that occur in type 2 diabetes. And this is highly interesting for us, and for that reason we really need to annotate the cell types properly and come up with ways where we can integrate the disease samples into the healthy atlas, reference atlas. And we are seeing that the interrogation of healthy pancreas is offering some surprises in the way of micropathologies, and we are really interested about exploring this further, trying to understand how these micropathologies could predispose to later disease. And with that, we are happy to take any question. Thanks a lot.
Thank you. I see a question over here.
Great talk. Can you talk a little bit about the ISS data? I think you talked about the codex, but I didn't get to see that.
So, to be honest, the ISS data is really just coming up. So the main focus was on the codex, and this is going on for a long time. And yeah, there was a lot of quality controls and so on, and we're just starting now to collect all the data for the ISS.
That's awesome. Is that also on the TMAs?
Yes. Yeah.
Very cool. Thank you.
Any other questions? I see nothing online. We have one back here.
Thanks for a really interesting talk with so many different modalities. Have you had first forays into trying to integrate the full-length data with the non-full-length specifically? And how has that worked?
Yeah, we have started the integration, and it seems that it's working quite nicely, and so it's quite promising. So now, of course, we will still face a lot of challenges trying to integrate the substates and of the different cell types, but so far it's looking promising. Yeah, it's too early to say.
With which method?
So I think that we have tested different methods, but so far Harmony is offering the best results for us. But I'm happy to discuss later if you have some other other suggestions. Thanks.
Any other questions? Oh, we have one down here.
I was very impressed by the number of lesions you found in healthy pancreas. I was wondering how old were the subjects you looked at?
So they're all not super young, not super old, right? They're all around 40, so I think 45 to 60 something is kind of the range.
Quite young.
And pathologists found kind of a maybe correlation between age and accumulating lesions, but yeah, we have to look into this a bit more.
One from Jonah.
Yeah, I was also intrigued by the microlesions, and I'm curious whether within a single pancreas, did you find multiple lesions of multiple types, or did they kind of like cluster where certain patients maybe had one or the other types?
So definitely, we actually find also different lesions in the same patient, and often. And I mean, since they're super small, I mean, sometimes 5 microns or something, you also find them in some slides, but they're not on the adjacent slides. So this will be also a challenge to bring all of this together, and then, yeah, maybe also have to change to really have these small lesions that maybe then develop into actual disease states.
Wonderful. I think, is that it? If we could give both our speakers another round of applause. So our final speakers for this session are Christoph Bock and Sabrina Ladstetter. I think it might just be Sabrina.
So, good afternoon, everybody. It's only been me today talking to you because Christoph is sharing anyway so much, so I thought you can leave the stage to me. I'm happy to present you the work of the consortium of the HCA organoid today, and And I will just start with, is it moving? No, I turned off. Sorry. Doesn't move. Now it does, okay. To just head up, we have heard about organoids, or the term organoids has been mentioned several times during the last days, and why is that? And also, why are organoids important for the Human Cell Atlas initiative? So we all are aware that those are models that can resemble human physiology and pathology, and therefore they are tractable and they are perturbable, and they will feed into the declared aim of the Human Cell Atlas to drive for biomedical impact, and they can be used to also study deeper biomedical fields like in the drug discovery or regenerative biology terms. Come on. Am I doing something wrong here? I could need some assistance. So maybe I briefly introduce in the meantime to you the consortium. So we are we started in 2020 with a project, and we got some funding. Thank you. From the European Commission, from the Horizon 2020 project, and we are in total 8 partners localized within Europe, and we are specialists in single-cell sequencing, single-cell imaging technology, and of course organoid technology, and we also get great support from the data analysis groups at the KFZ and the EMBL-EBI. So to just also give you a bit more background to human organoids, so organoids have been established for most organs of the human body already, and they do recapitulate aspects of human organ development in vitro. If we play it down, we can separate them into 2 types. So one is that they are derived from adult tissue stem cells. Those protocols are rather straightforward, reproducible, and relatively easy to scale. A good example for those are epithelial organoids, which are also derived for the gut or in the intestine. On the other hand, we can derive organoids from pluripotent stem cells, so either obtained from the embryo or by somatic reprogramming. Those protocols for organoid derivation involve multiple steps, are rather complex and slow compared to the ones derived from adult tissue stem cells. A good example here are the cerebral organoids that were already shown in detail by Barbara before. we still have a lot of key challenges when we talk about organoid technology, and also if we want to advance organoid research more to track down biomedical questions. So first challenge is that we have to characterize and really validate organoids as valuable models of human biology. And also based on this, we want to develop more organoid models as well as improved protocols that include diverse set of cell types. And at last, we want to apply organoids to basic biology and biomedical research, and all those key challenges can be addressed by generating an organoid cell atlas within the Human Cell Atlas initiative. The approach we are taking is a four-step approach. First, we generate an organoid resource that shall be shareable with the community. So in our pilot project, we do focus on only 2 organoid types for now, for the colon and for the brain. And we do derive those organoids from 100 healthy individuals each. From those we will generate, or we are generating reference maps using multi-omics approach to do single nuclear RNA-seq as well as single nuclear text-seq from the same cell. We complement this with bulk whole genome sequencing, and we also perform single-cell imaging on the organoids. All the data generated are fed into a computational resource that in the end shall be an interactive data portal for easy comparison of the organoid data to the primary tissue data. And we want, of course, to prove the value of our portal by performing disease-centric research. Coming to the organoid resource, so for the primary tissue-derived organoids, the colon, our partner, Hub Organoids, is collecting primary patient material and deriving organoids from those, and is generating that way a living biobanks that can be shared with the community. Our aim is, as I said before, to derive 100 organoid lines. Here I just want to mention that roughly 25% of those will have also matching colorectal cancer lines in the biobank. Currently, we biobank more than 80 lines already, so that's a good progress. And on the other hand, we have the pluripotent stem cell-derived organoids, the cerebral ones, which we do derive from already biobanked iPS cell lines under the HIPSCI initiative. And also there's the aim to derive organoids from 100 different cell lines, and we currently could accomplish growing 88 of those. There have been some considerations when we started to profile organoids. Of course, as with primary tissue, we have to consider the yield of cells that we can obtain from the different organoids, because they differ quite in size if you compare colon organoids and brain organoids. Also, the sample pre-processing to generate high-quality single-cell profiling data needed to be optimized. And what is also very important compared to primary tissue is that we need to work out a differentiation regime for the organoids to increase cellular heterogeneity and also to reflect better the in vivo situation. Here I just want to show you the example for the colon organoids, which are, if they are biobanked, cultured in an expansion media, where you will then in the end up with stem cells, transient amplifying cells, and a few early enterocyte cells. So it's not really complex if we compare it to an epithelium of the colon. So upon applying a specialized differentiation media, we obtain also enteroendocrine cells and goblet cells on top of the other cells. This was also nicely shown by Priska on Monday, who is our partner for the spatiotemporal high-throughput single-cell imaging of epithelial colon organoids. And there you just see again the snapshot of different days during differentiation of colon organoids and how different cell types do emerge. And Priska's lab is able to then generate trajectories of development, and currently they have already imaged more than 800,000 organoids from 57 donors. On the sequencing aspect, as mentioned, so we are using the multi-omic approach that allows simultaneously to profile transcriptome and epigenome from the same cell. The starting material for this is a nucleus suspension, so which gets in bulk transpositioned, and then using the 10x genomics platform, these transposed nuclei are fed into a microfluidics device to generate a gel bead emulsion that allows then single nuclear barcode attachment, and on top of this we perform then reverse transcription to to allow them to split the sample for gene expression library preparation as well as a text library preparation. And in that way, we get from the same cell, or in that aspect, nucleus, the single transcriptome as well as the single epigenome data. Also here we had the chance to get the hands on the multiome approach very early, so we were able to also compare the multiome approach to the standalone RNA-seq and ATAC-seq data. Here you see an example of QC plots for cerebral organoids, and overall the number of detected genes and the clustering is very similar between those two approaches. And also for the ATAC-seq we got, after integrative analysis, very good overlap. Just to briefly sum up the overall progress that we made up to date in our project. So we have 91% completed of our organoid resource, and as I mentioned, like 88% of the brain organoids. The single-cell sequencing of the multiome is also speeding up, so we are nearly at 80% for all samples we want to profile. Here, just also would like to point out that next to the organoids of the colons that we profile, we also have the chance to really also profile the matching primary tissue with a single-cell multiomics kit. And the single-cell imaging is lagging a bit behind, but we are ramping up a lot, and also our partners at IMBA, who are doing the imaging of the brain organoids, are doing this now at full speed and high throughput. But all the data in the end need to go into an organoid cell atlas, and for this we will perform data integration, and all the data are going to be placed or channeled into our HCA Omics Processing Center that is led at the DKFZ by Oliver Stegler's lab. And the idea is that the data are going to be deposited in public resources and then also fed in the HCA data portal. And we aim to launch the HCA organoid-specific data portal still this year. So in the end, I only would like to end with the overall vision of building an organoid platform to advance biomedical research. So this is just the start. It's just 2 different types of organoids, and it shall hopefully expand in the future to profile more organoid types, to validate organoids as space for robot models for human biology, and that we can develop better models. And in the end, we really would want to move forward to apply them for biomedical applications. And with that, I'm ending and taking questions. Thanks.
I see we have a question over here.
Really cool talk and great vision towards this. Do you expect an integrated organoid atlas to be integrated across different tissue organoids? Or is it multiple organoid atlases, each for different types of organoids? And how do you control the heterogeneity there?
So at the moment, the primary aim is to really integrate one specific organoid time together with the primary tissue aspect to really integrate across different organoid types is a good point for further work, but it's currently not set.
Got one here.
Very nice, exciting project. I was curious about the iPS-derived organoids. In principle, it should be easier to make that a diverse cohort because you can obtain iPS cells. They don't have to be geographically from your location, presumably. So is that a possibility?
This is definitely a possibility to do that. For now, we kind of limit it down to not get too much diversity because we are only N100, which is great, but we rather channel to get like a more like matching cohort to look at natural variation within those.
Sorry to push you on this, but what is too much diversity?
This is hard to tell for me at the moment because I am not so good in the bioinformatics statistics, admittedly. But I just know that for the selection of the lines we took, we did not especially really take the opportunity to do like 20% of this ethical background and so on. So.
I'm going
All right,
to move us
thank you.
on. We've got a question down here. And then,
Just a very quick comment to that. The more diversity, the better, but in this case, we needed open access consented lines for which we could put the full genetic information online. And they come from the HCA resource, and this is quite limited in terms of the genetic diversity that they have. So what we as a community urgently need is open access consented lines from other genetic backgrounds. This would be a very, very valuable resource that could allow integrating a lot of this type of genetic diversity into studies like this. But if they're not open access consented, which obviously is difficult if you go into vulnerable populations, then obviously you limit the usability of the data quite a bit.
I noticed that you also collected whole genome sequencing data for your organoids, which sounds really cool. I was wondering, have you already looked at that, and what scale of data do you think you need to really start linking, for example, genetic variants to phenotypes in your organoids?
So this is something that is planned. We collected only the primary material for that. At the moment, we did not yet start with the data generation. I'm sorry, so we don't have any insights there.
any other questions? Anything online? No. No more questions. Okay, another round of applause for our final speaker. Thank you, that was great. So that wraps up this session for now. We now have a half-hour coffee break. It's your last opportunity for pastries. So we will reconvene at 4:20.
Okay, welcome back to the final session. So the final session has the title Future Perspectives, and we have three very exciting talks here that try to open up in very different ways directions that the Human Cell Atlas could take over the coming years. And then we'll conclude with a few final remarks, and as a highlight, the announcement of where the next Human Cell Atlas meeting is going to happen in 2023. But now let's start with Jürgen Knoblich's presentation. Please.
So thanks a lot, Christoph, and thanks a lot to you and all the organizers for inviting me to come to this fantastic meeting and present our work here. Before I start, I would like to declare that I have two conflicts of interest. I'm a co-inventor on several patents related to organoid technology, and I'm a co-founder and advisory board member of AHEAD Bio. But what I'm really doing is I'm interested in brain development, but by training I'm actually a Drosophila geneticist, and what I would like to do today is I would like to convince you that in the time of organoid biology, we are making progress towards allowing an analysis of the development of the human brain using methodologies that were previously only available for invertebrate model organisms. But before I can do this, I need to give you a little bit of background on how the mammalian brain actually develops. So broadly speaking, in our brain, there's really two types of neurons. On the dorsal side, there are excitatory neurons that use the neurotransmitter glutamate and arise in an area that's called the ventricular zone and then form the 6 layers of the cortex. But there's an equally important second neuronal population that arises in the ventral parts and then undergoes long-distance tangential migration to integrate into the circuits of the dorsal cortex. Despite their differences, these cells arise from very similar progenitor cells. These progenitors are called radial glia. They initially divide symmetrically to amplify the progenitor pool and then switch to an asymmetric division mode where they generate various types of transit amplifying cells, and ultimately on the dorsal side the neurons of the different cortical layers, first the upper layers and then the deep layers, and in the end astrocytes. And on the ventral side they generate various classes of interneurons and then oligodendrocyte precursor cells. So a couple of years ago my lab has developed a method that's called the cerebral organoid method that allows us to recapitulate these developmental processes using iPS cell-derived pluripotent stem cells as a starting population. And while we know a lot about the different lineages that form the different types of neurons, the gene regulatory networks that are at the heart of these different cell fate decisions are much less well understood. And so what we thought we could do is we could perturb serially interesting transcriptional regulators in organoids in parallel, generating mosaic organoids where in each cell one of the transcription factors is knocked out, and then analyze the consequences of this on a single cell level. And as a starting set for this, we reasoned that genes that are responsible for neurodevelopmental disorders must have important functions during brain development. So we wanted to take a collection of disease risk genes, knock them out in parallel, and then analyze their consequences. And when I say we, I'm talking about Chong Li, who is a postdoc in my lab, and it's actually here in the audience, and all of this is a collaboration, a very fruitful collaboration with the laboratory of Barbara Treutlein. In order to do this, we developed the so-called CHOOSE system, which uses two genetic tricks. The first one is we start organoids with pluripotent stem cells that are transfected with verified guide RNAs targeting individual target genes, and we do all of this in the background of an inducible Cas9 line. And there's really two genetic tricks that we use in order to make this possible. First of all, the guide RNAs are inserted into the long terminal repeats of the lentiviruses so that two transcripts are generated. The first one for genome editing from the Pol3 promoter, and the second one from the Pol2 promoter so that it can be captured by single-cell RNA-seq. The second trick that we use is that the Cas9 is not expressed because there's a floxed stop cassette that separates it from the promoter, and this can be removed by a tamoxifen-inducible Cre recombinase. And the readout that we're using for this is single-cell RNA-seq. So for each cell, we can read the entire transcriptome, and we can read the guide RNA that it expresses and the genetic perturbation. And so the results of this are shown here. This is the single-cell map of an organoid. In this case, I've combined all the different perturbations, and you can see the two lineages. Here is the dorsal lineage. The radial glia cells are down here. Here are the transit amplifying cells, and up here you have the cells of the individual cortical layers. Here is the ventral lineage. The cycling radial glia cells are down here. Here are the cells that are more differentiated and dividing asymmetrically. And up here are the neurons that are differentiating into the different interneuron lineages, which are called the caudal ganglionic eminence and the lateral ganglionic eminence. So we use this system to screen through all the level 1 risk genes for autism. And we stratified those cells for transcriptional regulators. And the results of all of this is shown here. In this chart, I've plotted all the effects that these genes have on the balance between dorsal and ventral neurons, between excitatory and inhibitory neurons. And what you can see is there's some transcription factors that result in an increase in ventral cells. There are others that result in an increase in dorsal cells. I'll show you two examples here. ARID1B guide RNAs are depleted from the dorsal side, meaning that ARID1B is required for the generation of dorsal neurons specifically. ASXL3 is depleted from the ventral side, indicating that it's required for interneuron generation. So on this level of detail, we can annotate some loss of function phenotypes to some of the transcriptional regulators, but by and large, no common scheme is emerging. But what I would like to do for the rest of my talk is I would like to show you through guide you through a number of different, more sophisticated types of analysis that suggest that there may be a common underlying perturbation that is responsible for many of these autism disease types. So first thing we can do is we can plot the cells along their different lineages. So here are the progenitor cells, here are the transit amplifying cells, and then the different layer neurons. And one thing that we noticed was that there are common enhanced perturbations that are emerging from this. So for example, in the dorsal, but they're different on the dorsal side versus the ventral side, and that's why they have not been noticed before. So on the dorsal side, we find that very commonly there is a depletion of intermediate progenitors. Remember that these are the transit amplifying cells during the generation of the dorsal excitatory neurons. And on the ventral side, there's an enrichment of ventral radial glia cells. So generally, it seems like there is a block in the differentiation towards the interneuron lineage. What we also can do is we can group these cells and ask, are there several members of particular protein complexes that cause similar phenotypes? And one of them is the BAF complex, chromatin remodeling complex, where 3 members cause very similar phenotypes. And this will become important later in my talk. So depletion of intermediate progenitors on the dorsal side and enrichment of ventral radial glia cells on the ventral side is an emerging, seems to be enriched in autism risk genes, into perturbations of autism risk genes. Another type of analysis we can do is we can plot the cells along their lineage trajectories. So here we use RNA velocity to actually plot the cells along their lineage trajectories, and you can see the trajectories start at the radial glia cells, transit amplifying, and then the different cortical layers. And we can differently plot the cells along the different bins of these different lineages. We used gene expression in those individual bins to annotate cell types to these bins. So here you have the proliferating radial glia, progenitor cells, here transit amplifying cells, and here first the deep layers and then the upper layers. And when we plot all the guide RNAs collectively, patterns are emerging that you do not see when only analyzing individual genes. So there seems to be a common depletion of the guide RNAs from the transit amplifying cells. This is consistent with the previous depletion of intermediate progenitors that I've shown you, but very interestingly, there's an enrichment of them in the deep layers, and there's a depletion in the upper layers. So it seems like the transition from deep layer to upper layer neurons is one of the weak points of human brain development that makes our brain particularly susceptible to autism gene risk factors. And it's very important to note that it is this lineage transition where there is a high divergence in gene expression pattern between humans and primates. We also analyzed the genes on the ventral side, here we use a circular plot of the cells that are going into the individual different lineages. So here I've plotted the lineage transition probability. The progenitor cells are in the middle, and then the cells are plotted according to their probability to differentiate into the LGE, the CGE, or a different subtype of neurons. You don't need to know what these different regions are. These are regions that give rise to particular subtypes of interneurons that are characterized by particular expression of different neurotransmitters. There's another very interesting lineage, which is the OPC. This stands for oligodendrocyte precursor cells. These oligodendrocytes also come from the same regions, but of course they become glia cells, and so they are quite different. And in this case, we've decided to analyze only one of the genes, which is ARID1B, one of the members of the SWI/SNF complex, which is the one gene that actually caused the strongest perturbation on the ventral side. And when we ask what is changed in these lineage transition probabilities in ARID1B mutant cells, we find that there is an accumulation of the guides in this radial glia cells that go towards the LGE, but there's a strong enrichment of these guides in the oligodendrocyte precursor cells. This is highly statistically significant, and it tells us that in the absence of ARED1B, the progenitor cells are more likely to become oligodendrocytes than they are to become interneurons. But what we wanted to know is this conclusion, does this conclusion actually hold true when we analyze data coming from real patients? So we recruited a cohort of patients who carry mutations in the ARED1B gene. We generate iPS cells, and we use those iPS cells to generate organoids. And the outcome of this is shown here. This is a ventral region of ARED1B mutant organoids. And what you can see is that OLIG2, which is a marker for oligodendrocyte precursors, is very significantly upregulated in both of these patients. So number one, our organoid screen makes predictions that actually hold true in real patients. And number two, ARED1B is required for progression along the interneuron lineage, and in its absence, there is a higher likelihood to become oligodendrocyte precursors. Now, one important feature of this is that the interneurons that are generated on the ventral side, as I showed you on the first slide, they're migrating out, and that's a reason for why these ganglionic eminences are becoming smaller during development. And in ARED1B mutant organoids, there's a higher likelihood that they actually become oligodendrocyte precursors, which in this early stage are not migrating out. So one prediction of this would be that these regions of the developing brain should actually be larger in these ARED1B patients. And so we tested this using pre-existing fetal intrauterine MRI data. So there's, we were lucky in that our clinical collaborators had high resolution fetal MRI data, and we used that to do volumetric measurements. And what we find is that the ganglionic eminences indeed in those patients are enlarged, and this is happening throughout development. So our organoid screen can be used to make predictions which then actually hold true in real patients. Now, one other way that we can use to analyze our organoid data is we can predict target genes. We can predict genes that are deregulated in organoids that are mutant or perturbed for particular transcription factors. And we can do that in a regionally specific way. And so here I've plotted for you the different genes that are deregulated specifically in the dorsal and the ventral lineage. And what you can see is that, for example, there's a gene that's called CHCHD2, which is downregulated both in the dorsal and on the ventral side, and it's a mitochondrial protein. And we generally find that ubiquitin proteasome pathway and genes that are involved in Parkinson are particularly strongly deregulated. But what's really important is that when we ask what are all these deregulated genes, we find that they're highly enriched in other genes that are also in the Safari database of autism susceptibility genes. And this suggests that there may be actually a common regulatory network for autism. And in order to identify this network, we used an algorithm that was generated in Barbara's lab that allows us to predict gene regulatory networks. And we did that because we had multi-ohm data from these organoids as well. And doing so, we find specific gene regulatory networks for the excitatory neurons, also for inhibitory neurons, and for neural precursor cells. And what we now can do is we can actually ask which of these gene regulatory networks contain many genes that are actually deregulated in the autism-perturbed organoids. And this allows us to derive gene regulatory networks that are specifically susceptible to autism-like perturbations on the dorsal and on the ventral side. And one of them stands out, which is OLIG1, which is the most highly perturbed gene regulatory network. And it's actually very interesting that this is also a gene regulatory network that has been found in analysis of adult brains from autism patients. And so, although it's a bit too early to actually conclude this, there might be a common transcriptional signature that is responsible for this disease. So in summary, I hope I've been able to convince you that we can use organoids to learn something about human neurodevelopmental disorders that was not possible before. I've shown you that we've developed the CHOOSE system, that is a precisely controlled screening strategy in organoids. I've shown you that perturbation of autism genes causes imbalances in dorsal and ventral telencephalic cells, that there's a depletion of dorsal intermediate progenitors, enrichment on the ventral side, and I've shown you that the multiome analysis identifies dorsal and ventral autism-specific gene regulatory modules. So in the end, I just wanted to really acknowledge Chong Lee, who is the hero of all of this and has developed this methodology. Katerina Costa was the one who recruited the autism patients, and all of this is a collaboration with the laboratory of Barbara Treutlein in Basel, in particular Jonas Fleck, who is the person in her lab who works on this. Our clinical collaborators are Gregor Kasprian and Marlene Stümpflen at the Medical University. These are the organizations that fund our work, and I would like to thank you for your attention.
Thank you. We have time for perhaps 2 questions, one on the back. Please say your name and your institution.
Oh, Henry Jasper, Genentech. Jürgen, that was really cool. I was wondering, how unique are these outcomes for human neurons and organoids in this early developmental stage? Because you're recapitulating an early developmental stage, right? If you would do similar perturbations in the mouse system, do you expect similar outcomes?
This is a very difficult question because it hasn't been done, and I think so. In humans, the patients actually are heterozygous for these mutations. They're not homozygous, and for the vast majority of these genes, when you make them heterozygous in the mouse, not very much will happen. When you make them homozygous in the mouse, in some cases, there are similar things, but there's also other cases where the outcome is totally different. And it's generally known that the mouse brain is much less sensitive to genetic perturbation than the human brain. The vast majority of disease genes in humans that are dominant in humans are recessive in the mouse.
I have a beautiful work, and I have a bunch of kind of related questions. First, on your point of the in human, it's haploinsufficient. In the organoid model that you showed us, were these actually haplo, or they were actually doubles? Or you can't actually tell in the experiment?
Yeah, so I think this is a question I get very often, and it's a very, very important question. We have now we have actually collected nuclei to do single nuclear sequencing of the regions, which will actually tell us. I mean, we actually believe that most of them will be heterozygous, but I cannot tell you for sure, because many of them are cell lethal when they're homozygous, right? So we hope that actually, but I think it's a very, very important thing, and I think we need to do it.
And then my other question is sort of both on the when you hit with the mutation and also a canalization question. So if I understand the system correctly, it's inducible, and you introduce you activate the guide expression only at some point in development? Or?
The guide is expressed all the time, but Cas9 is induced at the neural.
Only at some point in the system. So do you have a particular sense of when to induce it, or just you do it when you trigger the iPSCs to start differentiating? So it's really at
we trigger it at around the stage of matrigel embedding, because we know iPSCs, you know, it's a cell type that doesn't really exist in humans, right? So we want to avoid artifacts from there.
So my main question is my canalization question. If you carry the organoids to later and later ages, do they still appear to be perturbed in the way that you would expect them, or did they manage to canalize some of the early disruption that happened in them? Because these were like 4-month-old organoids, right?
Yeah.
And they can go later, these organoid cultures.
They can grow later. There's work from Dan Gashwin's lab that has actually shown that even transcriptional signatures that appear only after birth do occur in organoids later on, so they do mature. I mean, by the way, this is much longer than you can culture neurons in 2D, right?
No, I know. My question is...
But do they adjust later on? I couldn't really say. We haven't actually looked. It might be very interesting to actually grow them for even longer, but these experiments are just, it's just very, you know.
With Paula, with Paula Arlota, we did an organoid experiment with 3 haploinsufficient, so I wouldn't suggest comparing to that. But we also did, for many of the ASD genes, we did an in vivo perturb-seq in the mouse, so that one
I know.
can compare maybe, but you can't compare because it's not the same age, right? The age of the organ.
Well, so the RED1B that you had in this paper is similar to this one, right?
Yeah.
Although I think there's information here that goes beyond that.
Right, absolutely.
And in the mouse, I think it is a bit, I think that the results are a bit different.
But it's also this apples and oranges of ages, but if you carry the organoids later, you would be able to do the comparison. That was
I think it's the combination of, I mean, I think we're all generating data sets that shine light from different angles onto the same thing. And I think in the end, we need to integrate them in order to arrive at common biological principles for this. Yeah.
So very quick final question, short answer, please.
We had a lot of fun, great talk. We had a lot of fun discussing during the break the potential limitation of the system. So I think coming from somebody like you, that will carry weight to tell people what you think the limitation of the organoids are. As we discussed, I believe that the small size, the lack of systemic and local cues for development limit the type of things you can actually notice in organoids, but I would like to ask
so
you.
I just need to be provocative here, because I get this question all the time, right? So nobody ever asks about the limitation of a mouse. The limitation of a mouse is that it's just not a human. Full stop. And
that sounds like a discussion for the coffee break.
Certain things that you... So I would actually restrict the analysis in organoids to those questions that you cannot address in the mouse. And I feel that the combination of the human cell atlas and organoid technology is absolutely unparalleled and opens up new avenues towards biology that were not possible before.
Thank you. The next... The next talk will be virtual by Barbara Engelhardt from Princeton University. Barbara, please go ahead.
Hi, I'm Barbara Engelhardt. I'm going to talk to you a little bit today about my group's methods, thinking about spatial genomic data. Thank you so much to the organizers for inviting me, even though I'm virtual. And I hope you guys are having a great time at the conference. Oops. So today I'm going to be talking a little bit about spatial transcriptomics and proteomic data. So these types of data have really opened up a lot of doors to study different spatial structures, organization, and function in tissues right now, thinking about how cells interact, thinking about what's intrinsic in the state of cells and what's influenced by the neighbors of cells. And I'm going to talk to you about some of the different methods that my group has put together to be able to start thinking through some of these problems and the analytics behind these problems. So spatial temporal models, in my mind, can be used with these data as a tool for understanding a number of things. First, we would like to study the spatial and the temporally resolved state of cells. In other words, if we were to talk about a particular tissue and a particular location in that tissue, we would like to understand what the state of a cell is in that location of a tissue at a certain time. And of course, it depends on whether that tissue is healthy or diseased. It depends on what the cell type of that particular cell at that location is. All of these things matter. However, we'd still like to be able to describe that distribution. We'd also like to quantify the intrinsic state of cells and the extrinsic state of cells. I'm cribbing this from Aviv Regev's definition. And the idea is really that the variance of the intrinsic state of cells is not affected by their local environment, where the extrinsic state of cells is that the variance of these, the state of these markers is explained by their context or their local environment. We'd also like to understand healthy and unhealthy interactions between cells. Healthy interactions, obviously, when things are going well, and immune cells, for example, are actively helping out keeping neighboring cells healthy. Unhealthy interactions are obviously when they're not. So in terms of what models can do in these situations, we would like to think about them as tools for discovery. And my work generally focuses on dimension reduction. Today, I'm going to talk to you about dimension reduction and alignment. These are all actually sort of types of dimension reduction, but the goal ultimately is discovery. The four things that I think about when I think of spatial temporal models as tools for discovery are, number one, to study spatially resolved patterns in cell state and tissue organization, really understand the spatially resolved patterns that we see. And that's going to be the first application that I tell you about. Number two is to quantify the variability in the cell state across the location, samples, and population. That'll be example number two I tell you about. Three is to predict the effects of specific interventions on spatially resolved cell state. I'm going to tell you about that in number four, and these are going to be very fast, I apologize. And number five, think about designing targeted interventions for perturbations to state or interactions. I'm not going to tell you about that, but that's ongoing work in collaboration with Alex Marzin's group at Gladstone Institutes. So the first method that I'm going to tell you about is something called non-negative spatial factorization. It's a method for spatially aware dimension reduction. The goal here is to perform spatially aware dimension reduction with spatial transcriptomic sequencing count data. So the idea is that we have count data. We have reads from some kind of spatially resolved sequencing. And I'm going to show you some examples of that. And we would like to, leaving them as counts, we would like to understand what patterns that we see in these data. We would like to do dimension reduction on these data. Go from, let's say, 10,000 genes to a handful of features, let's say 20 factors, where each of those factors represent some kind of spatially resolved pattern. I'll show you again what all this means. So the model that we came up with actually looks like a fairly standard matrix factorization model. So we're starting out with data XY, where Y is going to be our vector of expression. So let's say we have 5,000 or 10,000 genes here, mRNA values, and these are going to be counts. And the X are going to be, in this case, the pair of coordinates describing the location in the sample that we have for where this observation was observed, was identified. And so here we're going to describe this observation Y as a draw from a Poisson distribution with a certain rate parameter, a scaled rate parameter, scaled by nu. And nu is just going to be basically a function of the total read counts. And then lambda, which is going to be the rate parameter, is going to be a linear combination of some weights times a set of factors. And these factors are going to be exponentiated so that they're non-negative. These non-negative factors are going to be a draw from a Gaussian process. And the reason they're going to be a draw from a Gaussian process is so that we can encourage these X, we can basically make the factors a function of the locations. What this means is that we're going to, through the kernel over here, the covariance matrix, we're going to encourage the spots, the locations that are nearby each other to be in the same factor. And a few nice things about our model is it's a parse-Bates decomposition. I won't get into that. They're matern kernels, so they're not actually that smooth because a lot of the spatial structure that we see in tissues is not actually that smooth. And we have very fast variational inference with inducing point methods. This is work from a postdoc, Will Towns, in my group, who was successful on data market this year. So some examples of how this works on data. Here's an example from SlideSeq V2, mouse hippocampus sample, which has a fairly small field of view, but a very nice single cell resolution, a lot of observations. So here's 10 spatial factors that we have. You can see very, very nice examples of regions in the brain that these spatial factors identify elegantly. So two of my favorite here, number 8 is the dentate gyrus granule layer, where you can see it just highlights it almost perfectly. Whereas in number 10, we see the meninges, which is a very thin layer of cells separating different brain regions. And you can see actually it goes underneath this other brain region over here. So beautiful identification of these spatially resolved brain regions in this example. We can pull out the top genes associated with each of these factors, and we actually see pretty poor identification of the same regions. Often these are identification of a particular cell type that's enriched in these regions, but all of this to say that actually single gene markers and small subsets of gene markers are often not sufficient to identify specific brain regions or specific cell types, in fact. So we often need a large number of gene markers. Here's two examples of non-spatial factors that we identified. So we also obviously applied non-spatial matrix factorization, and you can see that we pull out really nicely identified cell types. So here's astrocytes and here's neurons. We identified them just looking at the genes, but there's no spatial organization here, only in the sense that there are regions in the brain where astrocytes don't exist in the same proportions. So you can see a little bit of structure here, but it's absolutely not the same as the spatial structure we saw in the spatial factors. The next example that we looked at was this 10x Visium data. It is a much larger field of view, and it's not single cell resolution. So every single point, every single spot is going to be an average over multiple cells. This is also mouse brain data. We've labeled the brain over here with the help of a mouse neurologist. So here again, we see a really nice description of different brain regions with 10 different factors, latent factors in the mouse brain. So for example, we see in number 7, we see the outer olfactory bulb over here. In 9, we see multiple different brain regions over here. 5, we're going to see the olfactory granule layer really nicely highlighted there. And just as before, if we pull out the single top gene associated with the weights of each of these factors, we see good but not perfect delineation of each of these regions again. One thing that we can do with this non-spatial model is look up the spatial importance score. So as I said, we had spatial and non-spatial factors, and we can just divide by the variance explained effectively in the genes, and also the spots or the observations, cells if it's the single cell, or groups of cells if it's the wider field of view versions. And we can look to see how much spatial variation versus non-spatial variation. It explains each of these genes or the cells or observations. And you can see that there's actually a pretty large variation, and actually the distribution looks quite different for the cells and the genes as well, where there's a lot higher variation for the genes, I think not surprisingly, than the cells, which appear to be pretty locally variable relative to the genes. I'm not going to, bet money on this. I think there's a long way to go in thinking about variability, intrinsic versus extrinsic variability, but this is sort of our first pass estimate of how we're going to start thinking about this. The next example I wanted to tell you about was de novo alignment of spatial samples. So the idea here was to align different spatial genomic samples across different field of views, different technologies, different marker sets, and different resolutions. So here's an original sample, as you saw before. This is mouse brain again. If you warp it, we would like to align all these samples to the same, to a common coordinate system. And we'd actually like to do this without knowledge of the common coordinate system beforehand. This is work by my amazing PhD student, Andy Jones. So right now, a lot of groups take these samples and analyze them separately. But when we analyze them together, we can start studying variation in expression or tissue morphology. We can think about variation in the interactions between the cells and variation in cell type heterogeneity. So basically variance, which we can't really get when we think about these different spatial samples separately. So there's two sources of variation, not surprisingly, that we'd like to think about. So technical distortion, which is basically distortion that's due to tissue preparation or handling or sequencing, we'd like to get rid of that completely. So that's a lot of what the alignment does, is remove technical distortion. The biological distortion, we'd like to essentially partition. We'd like to identify all the distortion inherent to biological variants, and then we'd like to find the sort of partition of that variance that's interesting to us, whether it's due to disease status or sex or a particular intervention. That's what we're interested in looking at, variance due to something. So the model that we came up with, as is pretty standard for my group, is a deep Gaussian process. So we actually have two layers. So the first layer takes the different samples that we have of the spatial genomics data, and it warps them onto a common coordinate system. And the second layer is another Gaussian process that takes each point in this common coordinate system, each XY or XYZ point, and maps it to a high dimensional description of the markers. Those markers can be genes or proteins or even histology pixel values, really whatever you want. So whatever output you want here. And as I mentioned before, but I'll say it again, the common coordinate system can be a latent variable. It doesn't need to exist beforehand. So the idea here is that basically we want to identify this latent variable such that we can explain this description, these markers from each of the samples as coherently as possible with whatever this latent variable system is. So here's an example. It's just a simulated example with a single gene. So here's our observed spatial coordinates, 1 and 2, and the pluses and the dots are two different observations. And then when you align it, you can see here there's a lot of weird warpings and it's nonlinear, but you can see that in general, the warping does a really nice job of overlaying the different points where the expression values of the gene are nicely aligned. So here's an example of 4 parallel slices of a breast cancer biopsy from spatial transcriptomics data. This is from a Stahl et al. Paper from 2016. The first thing that we did was we aligned all of these 4 samples into a two-dimensional space. Here's actually the observed data space. So this is when we look at the X coordinate system. This is what the alignment looks like. This is our reference space. So this is our de novo alignment that we identified. If you pull out the latent structure, this is what that looks like. We also tried to align it to a 3-dimensional space. So we know the order of the Z-axis for these 4 slices. So we actually just pretended that there was a Z-axis and aligned those 4 slices according to that Z-axis. And this is what the X versus Z and the Y versus Z alignment of those 4 slices look like. And what we could get from this is actually what we're calling an atlas. So here we have the expression of a single gene, and here's actually a 3-dimensional tumor, breast cancer tumor for this patient. So we can actually query, because this is just a de novo alignment of these 4 slices, we can query any XYZ coordinate within this tumor for any gene. So we can impute at any XYZ coordinate just because these are a bunch of Gaussian processes. That's what they do. That's what they allow us to do. So yes, so because of this deep Gaussian process, this is what I think about when I think of an atlas. And we're hoping to do this basically for as many things as possible right now when we have multiple slices. We're actually working on pulling together mouse brain and a bunch of other examples of this with both gene and protein samples. Another example that we actually published in 2021 is live cell imaging data. So here, the blue examples, or the blue cells, are low ERK signaling, the green is high ERK signaling, and the red is just background. This is actually canine kidney samples that they've made a wound in. And you can see that as the cells go and try and repair that wound, you can see a lot of bursting going on here, trying to recruit cells to the wound frontier. Not a lot of bursting going on here as the healing is going on. So there's definitely spatially resolved signaling going on in ERK bursting. So we actually, in this example, wanted to understand the pulsing rates of ERK. We actually wanted to separate the pulsing into 3 different things. Number 1, the spontaneous pulsing rate. Number 2, the self-excitation. So when a cell pulses, how does that affect its future pulsing? And number 3, sorry, how does a cell pulsing affect its neighbor? As you can see, there was bursting related to neighboring cells. And we were able to do this really nicely. And in fact, we were able to do it in a spatially resolved way as well. So here are these dotted lines in each of the images are the location of the wound. And you can see that in these spatial bins, these represent distance from the wound. You can see that the base rate of pulsing and the cell signaling changes pretty drastically. DMSO is the control. So you can see that it basically base rate of pulsing starts out really low and then kind of goes up and then goes back down to sort of steady state. Similarly with cell signaling, kind of goes up, shoots up, and then kind of goes down to steady state. What we didn't know before we actually did this is that these 2 signaling repressive drugs actually repress the base rate of signaling, of pulsing of ERK. We didn't know that beforehand. We did know that they repressed the signaling. We didn't know that they repressed the base rate. The final thing that I wanted to tell you about, is phenotyping cells and live cell imaging. So this is what we're working on right now with Alex Marzin and Sidhu Jena. We basically have CRISPR screens of CAR T cells and 382 well plates. And our goal is basically to phenotype CAR T cells from tumor cells and then experimentally design these CAR T cells. I should say that another thing we're very interested in is thinking about using not just spatial information, but also cell type information to do dimension reduction. The way that we're going to do this is by using these multi-group Gaussian processes that we developed, and we have a paper on arXiv about this, where we can actually include categorical variables. These categorical variables can be the cell type. So we want to be able to include cell type in these dimension reduction methods that we have. And finally, another method, we're working a little bit with Jason Von Rostro and others on this, thinking about identifying QTLs in single cell data, because a lot of the methods that we have are not really working. So we just developed and put up on bioRxiv a telescoping bimodal latent Dirichlet allocation model for that. So a lot of work going on in our group, and I'm really excited about everything. So here's my acknowledgements, and here's a bunch of papers, preprints, and code that we have. Thank you so much.
Thank you. We have time for 2 questions, maybe.
I was wondering, you have a Gaussian process to model the spatial and gene expression relationships. Are there situations when that doesn't work? Like, when does your model not work, and do you know why?
That's a good question. we haven't seen a situation where it doesn't work that well. So the way that we tested it out of sample was we actually removed a whole bunch of locations, the gene expression at a bunch of locations, and then tried to impute them. And if you look in our preprint, you'll see that actually our method is extremely well in that. But again, versus there's always going to be some error in imputation. So just because of natural variation anyway. So when does it not work? That's a good question. I'll think about that and be very honest about it. But so far, it's been pretty good.
Thanks.
Hi. Great talk, thank you. I'm Lohan Vulia from the Max Poets Labs and the Center for Molecular Medicine here in Vienna. I have a question about your spatial dimensionality reduction technique, because you showed us the top genes that were associated with the different factors, and I'm wondering if you already looked not only at the top genes, but at all the genes that had a high weight to see if they corresponded or if they were associated in some ways that were either already known or new, and that would underlie the identity of these different regions.
Yeah, that's exactly right. So that's how we actually ended up identifying the regions. So we looked at the top genes. They sort of forced us to see specific cell types, and then we were able to label each of the regions based on those cell types. So again, in the preprint, there's a list of the top genes and also the cell types that each of those, that set of genes corresponds to. So yes, absolutely. Again, with these linear models, it's fairly easy to do that sort of thing. Yeah.
Thank you.
Thank you. Quick final question in the back. Okay, use the time to ask your question, please.
Okay, this is very confusing. So I should ask my question? Okay. Barbara, this was beautiful. I'm actually following up on the previous question. You showed us that in the spatial data, you showed us that the cell types are quite generally distributed. I won't use the word uniform, I'm not sure it was. And that you still had the regional signal, and you now told us that the regional signal did come from cell types. That's actually how you identified. So when you take the regional signal and you apportion it for the cell types, can you say something about, say, astrocytes in this location do this versus the general, astrocyte identity, which is not spatially dependent? Because that would be the intrinsic versus the extrinsic.
Exactly correct. So yeah, we actually found, for example, astrocytes that are very, very specific to single regions of the brain versus astrocytes like you saw in the non-spatial factor that were spread out everywhere. So I do believe, like I think you're hinting at, that there's going to be subtypes that have specific functions and also have specific cell type markers as well within specific brain regions. Absolutely. Yes.
Hi, thank you for the talk. I'm Andrea Gendair from SAM here in Vienna. I was wondering, for the Gaussian process-based method for image registration, whether it also works well due to the uniform grid of the Visium data, and whether you've tried the same approach on other types of spatial transcriptomics.
Yeah, so we tried it on Visium, SlideSeq V2, actually also XYZeq, and we even tried it on some protein, spatially resolved protein data as well. So far, it seems to actually be very effective for all of them. We've actually also, I should say, also done it on some fairly recent MERFISH data as well. And so far, so good. So Gaussian processes are nice in that when the locations that we're sampling aren't evenly distributed, aren't uniformly distributed, it does a very nice job of smoothing across the space where there aren't observations, but also allowing pretty big errors in imputation, in distribution. So that's one of the reasons actually I like it a lot, is because it sort of says, here's a desert of observations, and we're going to pretend we know what's going on here with these smooth functions, and we're also going to say we're uncertain about those places as well because there's a desert of observations. Yeah.
Thank you. Great presentation, nice discussion. Let's move on to the final speaker of the session. I would like to introduce Karuna Ganesh of the Memorial Sloan Kettering Cancer Center. Please.
Thank you all for staying till the end and for this amazing opportunity to talk about our work today. So I'm a physician scientist, I'm a GI medical oncologist and run a research lab studying stem cell plasticity during tumor progression with the goal ultimately of making an impact for our patients. These are my disclosures. So we study metastasis because metastasis is the reason really why most people die from cancer. And work over the last couple of decades, tumor sequencing efforts, matched primary tumors and metastases from multiple patients, have shown us that there aren't, by and large, many genetic alterations, specifically single gene alterations, that are selected for or that emerge de novo during metastasis. And indeed, tumor progression and therapy resistance are often problems of plasticity, where cells acquire properties as a consequence of non-genetic adaptations, whether they're transcriptional, epigenetic, metabolic, etc., to the stresses, the distinct stresses of the invasion, metastasis, dormancy, immune evasion, proliferation, therapy resistance cascade. And it is this plasticity that we're particularly interested in. And we think that addressing this plasticity may provide us with more opportunities to better target these tumors. This has really been limiting in the sense that we really don't know what is happening during metastasis. Single cell efforts are now just beginning to start to look at metastasis. And to date, there isn't any published data really looking at, in multiple patients, the transition from normal tissue to primary tumor to metastasis in individual patients and across cohorts. And this has been a real gap in knowledge for us. And the second part of that is, of course, once we know this, how do we dissect this and get to conserved mediators that one day we can therapeutically target? And for this, of course, we need good models. The mouse, as we've heard from Jürgen earlier today, is not the human. And part of the problem, if you like, with the human is that we're heterogeneous. Patients are heterogeneous in many, many ways. And unless we really address that heterogeneity and this plasticity, we are unlikely to be able to find truly conserved mediators that are amenable to therapeutic targeting. So these are some of the questions that we aim to address. And the way we've approached this is to establish in our institution a patient-derived biospecimen platform from patients with metastatic colorectal cancer. And the reason we study colorectal cancer is because this is the only common solid cancer across the board where once patients get metastasis, surgical resection, surgical removal of both the primary tumor and part of the normal colon and the metastasis at the same time is part of a standard of care for many patients. And this gives us really this very powerful opportunity to profile with all of our favorite technologies, single-cell RNA-seq initially, and also spatial technologies and other technologies, the normal primary met, sometimes lymph nodes as well, from the same patient, and at the same time derive organoid models from the same tumors and increasingly more complex model systems incorporating other components of the tumor microenvironment, including from peripheral blood where we collect PBMCs, as well as from stool and other microbiota from the same patients. And so our first attack, if you like, at this problem was to start to compile a single-cell atlas of this transition from normal to primary to met across cohort of patients. And our initial cohort at the moment that we've analyzed together with Donna Pear's lab contains 90 data sets from 31 patients. This includes about a third of patients who had not previously seen any treatment. So this is, if you like, the ground truth of metastasis. And then the other 2/3 had received standard of care 5-fluorouracil-based chemotherapy prior to the surgery when we got their tissues. In 6 cases, we were also lucky to have a metachronous metastasis, a secondary met that developed 1 or 2 years after we got the initial collection. So we can actually see tumor progression in time also after therapy. So what do these data look like? We see excellent representation. This is about 150,000 cells from these 90 data sets of all the major cell subtypes, including immune cells, stromal cells, and perhaps most importantly for us, the epithelial cell compartment, which really has not been very well studied or defined because of technical challenges in actually processing and handling this tissue well and getting high quality data, which took us honestly quite a long time to optimize and get right. But when we hone in on the epithelial cell compartment specifically, we see that across these 31 very diverse patients, we have the non-tumor data sets that actually overlap very well. Despite all the differences between patients, the normal tissue actually looks quite similar, which is very encouraging. And then we see the primary tumors and the metastases sort of clustering away from the normal tumor normal colon compartment. By labeling these cells for nearest neighbor-based entropy, we can identify subsets of these cell populations that are conserved. The brighter colors are high entropy. These are cell states that are conserved across multiple patients, which is exciting to see, and perhaps offers the best opportunity for eventual therapeutic targeting, as well as some blue populations that are unique to one or two individual patients. And when we look more closely, we have to sort of think about this in the context of what we know about intestinal development, of course. And we know that the homeostatic stem cell of the intestine, from the work of Hans Clevers and many others, is this LGR5 cell that lives at the bottom of the crypts in the intestine. And these LGR5 cells are constantly proliferating, dividing and differentiating to give rise to all of the different cell types of the intestine, including the two main lineages, the absorptive enterocytes and the secretory goblet cells. The LGR5 cell has also been shown to be the cell of origin for colon cancer, the cancer stem cell. But whether this stem cell state is preserved, and if so, in what capacity in metastasis, is really a big question for the field that has not been resolved, especially in patients. So when we look at our human data set, we see that the pancolonic differentiated cell state marker CDX2 is expressed along really in most of these cell states, in the non-tumor as well as in the tumor. In the non-normal colon populations, we see discrete populations of cells expressing either the enterocyte lineage or the goblet cell lineage, again showing us the high definition in this data. And when we start to look at the tumor, in the primary colon states, we see, as we would expect, a strong selection for LGR5 ASCL2 positive stem cell populations. These are not exactly the same stem cell states as we see in the normal colon, but they're clearly strongly related and still retain the expression of some of these markers. In metastasis, we see that there's a partial overlap with the primary tumor. These LGR5 cell states are still present, but now we start to see cells that sort of are at the edges of this UMAP. They're acquiring new states, often marked by dedifferentiation markers like SOX9. So to gain more insight into what these new states were, what was happening in the transition from primary tumor to metastasis, we applied an algorithm developed by Nir Yosef, published recently, called Hotspot, where we basically looked at co-varying gene expression modules across this entire data set. And we identified through this analysis 32 distinct modules of gene expression. In particular, we see this highly correlated variation of gene programs at the top left of this graph, which is deeply anti-correlated with this second population in blue. And when we look at this in more detail, we see that we can basically rank all of these gene expression modules on the left along an axis, sort of mapping them to whether these states are similar or different from the cell states found in the normal intestine. And when the states are more similar to the states we find in our normal colon data sets, we call them canonical. These are canonical intestinal states. And when they are quite different, we call them non-canonical. These are non-intestinal states. And we see that co-varying population of gene programs, the modules on the top left of that graph, those were all these non-canonical modules that are really never seen in the normal colon. And across this spectrum of states, we see a transition from the canonical gene programs on the left into an LGR5 stem cell population sort of in the middle as an intermediate state, and then a transition again into this non-canonical set of gene programs. And when we look at all the clinical variables that we know about these patients, because we have full clinical annotation and information about these patients, we see that the two things that correlate most with this transition from canonical to LGR5 to non-canonical are firstly metastasis. So primary tumors are much more canonical than non-canonical, and metastasis is strongly associated with these non-intestinal programs. And the second is therapy. So treatment with chemotherapy is associated with a further progression into these non-canonical states. And we can also map these states onto what's called the consensus molecular subtyping, or CMS classification of colon cancer, where CMS3 is the most canonical appearing and has the best prognosis. This is based on bulk RNA-seq of tumors. And CMS4 has the worst prognosis. And we see that CMS4 is strongly associated with these non-canonical states, but much more heterogeneous than had been appreciated before. We can rank all of our patient samples from individual patients along a spectrum of canonical and non-canonical. And so we can see that the more treated and more metastases the patients have, the further along the spectrum that they are. when we look at individual primary metastasis pairs from individual patients, we can map their progression along the diffusion coefficient that best maps the transition from this canonical to the non-canonical state. And by doing this, we can see in every single patient we look at, we see that the primary tumor is more canonical, the metastasis is more non-canonical, although clearly the extent to which this transition has happened does vary from patient to patient. And in those patients where we have a secondary metastasis in green now, we see that those secondary metastases removed 1 or 2 years after the first one was removed, patient has had further treatment, these secondary metastases are now even more non-canonical. They're further along the non-intestinal trajectory. So to learn more about how this might be happening, we actually turned to the question of the fetal programs through the HCA Fetal Gut Atlas from Sarah Teichmann's group. Sarah's work has, of course, mapped beautifully the gene expression programs in a variety of organs, in this case, in the fetal gut in the first trimester, where these cells are in what they call a uniform progenitor state. The endoderm differentiation is not yet complete. And the second trimester, where now major organogenesis is largely complete, so the gut is much more mature, though not quite intestinal. And by mapping these fetal first trimester and second trimester programs onto our gene, our diffusion components that map the canonical, non-canonical trajectory, what we see is that the second trimester signature, which is here in red, goes down along this trajectory. So the cells are getting more de-differentiated. They're losing intestinal identity, and they continue to go down along the trajectory. Whereas the first trimester signature, the fetal endodermal progenitor signature, that actually rises, it peaks, and then towards the end of this trajectory, it starts to go down again. And when we look at what's happening in individual tumors, this is a single liver metastasis from one patient, we see this entire spectrum of states. On the left, you see all of the canonical intestinal states, starting off with an intestinal stem cell state, which can then differentiate into all the different trajectories. That then sort of moves into an injured state, where we can map gene programs associated with regeneration and wound healing. And then from there, we then see this fetal progenitor state. And finally, we start to see non-canonical, non-intestinal, differentiated programs. In this particular tumor, we appreciate gene programs associated with squamous cell carcinoma. So these are keratins associated with skin differentiations or lineage plasticity that is starting to happen, and also gene expression programs associated with neuroendocrine differentiation. We can, of course, validate these hypotheses using a different approach, in this case using multiplex immunofluorescence on protein from the tissue from the same samples that we profiled using single-cell RNA-seq, and we see, of course, in the normal colon, beautiful intestinal crypt architecture, single layers of epithelial cells expressing CDX2. In the primary tumor, an expansion of a differentiated CDX2 positive intestinal cell program. But in the metastasis, we see far fewer of these cells express CDX2, the intestinal canonical lineage marker. And instead, we now start to see expression in blue of de-differentiated SOX2 stem cell markers, and also in red, we start to see expression in pink, of chromogranin A, a neuroendocrine marker, and in the bottom panel, we see slower proliferation, loss of Ki67, and a gain instead of keratin 5, which is a canonical skin squamous cell marker. So the questions from this are, we are seeing de-differentiation and transdifferentiation. How much of this is tumor cell intrinsic, and how much is driven by the different microenvironment of the liver? And based on this, can we identify conserved mediators that might be shared across multiple patients, because that, of course, would then suggest potentially opportunities eventually to target this transition. So to address this, we turned to the organoids that we'd made. So we made organoids from about half of the samples that I've shown you. And when we look at trios of normal colon, primary tumor, and metastasis organoids made from the same patient, and in this case, of course, now all of these are now put into the same media conditions. This is all human intestinal stem cell media. So in the same media conditions, in the same microenvironment, we see that they retain distinct morphologies. The normal colon has a cystic architecture, as has been well described, single layer of epithelium surrounding a lumen. In the primary tumor, we start to see some cells that still retain that cystic morphology, but many more that become, that have luminal filling, dense, multi-layered structures. And in metastases, we pretty much never ever see the luminal structures, and they're always just these dense structures. When we do single-cell RNA sequencing of the organoids from the same tumors that we sequenced, we see that, again, despite being in the same media conditions, they retain distinct gene expression profiles. And indeed, we can see that the primary tumor and the metastases, especially the metastases, has a strong selection for this fetal progenitor program and then for discrete regenerative squamous and neuroendocrine programs. We can map directly the tumor and the organoid states onto one another, and we see that despite the loss of phenotypic volume, cell state heterogeneity, by putting them all into the same media, the metastasis organoids do in fact retain many of the cell states, including the non-canonical states that we can find in vivo, although there's clearly a selection and expansion for the LGR5 intestinal stem cell state in organoid conditions, because of course that is what we're giving them in the media. So can we use the information from the single cell sequencing to do better? So first we wanted to know whether the organoids retain the potential to go to have different functions. And so to do this, we took the primary tumor and metastasis organoids from the same patient, transplanted them into the colon of the mouse, and found that both grew reasonably well, the primary perhaps a little bit better. But if we now take the same organoids and transplant them into the liver, the primary tumor organoids don't grow very well at all, but the metastases organoids grow much, much better, suggesting that they retain distinct potential for metastasis. And then now when we go back to the organoids and withdraw the growth factors that are intestine specific, we find the normal colon organoids die. The primary and met organoids will survive but not grow. But if we now replace the media with other factors that we now find from the liver, now we can find that the primary tumor organoids don't grow very well, but the metastasis organoids absolutely can adapt and grow. And depending on what we add to the media, we can push them in different directions. They have tremendous plasticity. They can go into goblet cell differentiation directions, into enteroendocrine or neuroendocrine differentiation directions, and if we treat them with chemotherapy, we can get them over the canonical to non-canonical hump, going through that injury repair cycle, and then we now start to see the gain of a fetal progenitor program, the loss of intestinal programs, and the gain also of squamous lineage programs. Now, using our gene expression data for multiple patients, we've identified a bunch of transcription factors that are shared across multiple patients, which we are now performing perturbation experiments on to dissect mechanisms of transdifferentiation and finding out ways to block this. So to summarize, we've identified that colorectal cancer metastases span an expanded range of transcriptional states, and they have a cell-autonomous epigenetically programmed plasticity. We're dissecting mechanisms, enabling the selection of distinct states by microenvironmental cues. And epithelial injury is important for this entry into non-canonical states. The next steps for us in terms of the future are to now start to look at tumor microenvironmental coevolution by starting to look at what's happening to the tumor immune microenvironment and to the stromal microenvironment during this transition, modeling this ex vivo from the same patient samples using organoid, immune, and microbiome co-cultures, and iterating really with the single cell atlasing, organoid modeling to improve the way in which we can model these states using human samples, and then of course using perturbation experiments. So the ultimate goal really and the future is to use the patient as the primary experimental model to really understand and target the heterogeneity and plasticity of cancer. And this is all, of course, only enabled and possible because of these amazing reference data sets, including the ones like the fetal gut atlas that I just showed you. The conceptual and computational innovation that people in this community are making to enable us to access and understand this data, and the interdisciplinary collaborations we're developing. So I'll just conclude by thanking Donna immensely for all the computational work, Andrew Moorman in her lab leading the computation, and Francesco Cambulli in my lab leading the experimental work, and all our funders. Thank you.
Thank you. Questions, please.
Yeah, beautiful talk, Karuna, and thanks for acknowledging the data on the fetal. I was curious, like, is there, and I was a bit surprised that you didn't mention the EMT, kind of, because I would have expected that to be a major component of the transition to metastasis.
Yeah.
So is that there, but you just didn't mention it, or it's not there, or?
Yeah, I think, so EMT has long been controversial in colorectal cancer, I think I can say that. I think EMT falls under this larger umbrella of plasticity, right? It's a particular form of plasticity. Yeah, totally. And certainly we see, modules that have higher expression of STING, of twist and other EMT as a slug and snail and these associated genes, but it doesn't seem to be a dominant sort of striking program per se. I think EMT is part of this, but it's not the full story.
And then I guess one quick, that's fantastic, and thanks for clarifying. So one quick question, you were saying on the tumor microenvironment, you were showing the signaling on, from the T cells and so on. And I guess, I know you said that it's the next step, but for me, like the obvious question then is, you're saying it's epigenetically encoded, this transition. And so, like, what are the signals that are promoting that? Are they coming from immune cells or?
Yeah, no, those are great questions. Our best guess so far is, and this is work that we published previously, is that we think that while signals from the microenvironment are certainly important, the key trigger is really the loss of epithelial integrity. When epithelial barrier organs like the gut lose contact with their neighbors, the normal response should be to die and also to immediately sort of create a new barrier, otherwise there's no organism anymore. And so we think that that stress of loss of contact with their neighbors is necessary and sufficient to promote this plasticity. And we can actually show this by just taking intact organoids and dissociating them into single cells, or by treating with chemotherapy. Any way you damage contacts between neighboring cells, you can induce plasticity. But then, of course, with serial selection, you select for these states that are kind of almost permanently in that way. And we're figuring out more about mechanisms.
Hi, just here, Musa Hanifa. Thanks very much, really enjoyed it. I actually have a question that's slightly related. Do you think, because it wasn't clear to me from all the evidence that was shown, that actually that acquisition of plasticity was critical for metastasis, or that you have this proportional representation of greater cells with plasticity in the liver simply because of the microenvironment, i.e. they don't really have the sort of same epithelial contact, etc.?
So, I mean, the way I guess we've addressed that is by putting them into the same microenvironment ex vivo, right? But even in vitro, they retain, so the primary tumor and metastasis organoids have distinct abilities to adapt to different growth factor conditions.
What I mean is that do the metastatic cells, do they have to actually acquire this program before they metastasis? Does this actually confer greater metastatic potential?
Yeah, that is a great question, and that's something that I think we'll have to experimentally test, because now, of course, we're only getting what's at the end, right? So we don't know how they started off. Yeah.
Thank you.
Hey, Henry Jasper again. A very nice talk. If you would go to a different site of metastasis, this is all liver metastasis, right? If you would go to a different site, do you expect to see a fundamentally different signature of plasticity, or are they going into probably the same state?
Yeah. So our data set is primarily liver metastasis. We do have a couple of tumors from other sites, and we would love to address that question. I'll tell you preliminarily that from the lung, we see a different sort of plasticity for the most part. They tend to actually be more goblet cell-like. I have no idea why, but we don't have enough statistical power to make great statements about that yet. But I'll also tell you clinically, I have a lot of patients who have lung mets, but nobody ever dies of lung mets. I mean, almost never, because they tend to be very small. From colorectal cancer, they tend to stay small. They don't really cause clinical problems. The liver met is what causes problems. So maybe there's something there that they're staying very canonical there. I don't know, but this is all speculation.
Thank you.
Thank you.
So that brings the scientific part to a close, and in the final session, we would like to wrap up very briefly together with Sarah and Aviv. Thank you very much. This was a great conference. This is the group picture. We will share this in some way or another with you. There's also quite a lot of documentation on Twitter, so if you haven't looked at, here's the hashtag. You will find a lot of excitement and reporting, especially the speakers here. You will see a lot of great feedback regarding your talks on Twitter. My personal highlights of the conference, first and foremost, that we all meet again in person, and it was really nice. It's this full body experience of eating, drinking, and seeing, and touching, and these type of things. So until I think we are all kind of avatars that we can just beam ourselves to some places and are purely virtual, I do think there is a positive
also economic footprint ratio or environmental footprint ratio of traveling at least sometimes, because based on the discussions I have here, I'm much more effective in doing the right things. And if I just don't start a project that's redundant or team up for a project with someone else, I save so much of a carbon footprint compared to just doing my kind of narrow-minded thing without talking to other people. Obviously, we're taking a bit of a risk here with COVID. I see some masks, I also see some non-masks. Can you please, if you turn COVID positive the next 3, 4, 5 days, still let us know. I would like to do, just as a mental exercise, know how many infections might have arisen from this conference, just to get a feeling for how much of an, not only carbon footprint, but kind of epidemiological footprint we are leaving here. So I saw a lot of great science. This was really nice. Highlight of the conference, definitely, every talk. The atlases are coming together. The roadmap is there, developing. The biological networks were, I guess, a really good invention. That's working. And now we are all excited getting this white paper 2.0 together. I personally liked how a lot of people contributed, and a lot of people that were here for the first time that I have not seen in the human cell atlas context before. And in particular in the breakout sessions, a lot of unplanned things happen. And I think this is what the breakout sessions are for, giving room for cool ideas to develop. And I hope some of these will find kind of a group of people that will take these things forward. And finally, there's a lot of hard work ahead. I would in particular like to also thank those people who are critical and who ask hard questions, highlight problems, etc. Because there is kind of the stuff that we're doing is not easy. And sometimes a quick and dirty solution might be easy, but the right solution and getting the atlas right, that's hard. But it seems that the community is there and can do it, and we just need to force ourselves to the rigor and precision and to actually do it. So these were my 5 take-home messages. I think many of you will have their own. That will be very interesting to hear, actually. I would conclude this kind of my part by thanking all the participants. We had quite a global distribution here of the people attending, especially virtually. We had 220 people in person, 641 that logged in live for the virtual thing, and in total 1,500 who signed up for the system, so who might watch recorded talks. Actually, this talk recording is really good against fear of missing out, because I don't like breakout sessions because I have to decide. But I do like recorded breakout sessions, because afterwards, if I feel like I missed the regenerative medicine breakout session today, which I really regret, I will watch that at home. Thanking the sponsors, a lot of sponsors contributing, Jan Zuckerberg Initiative, Klarman Family Foundation, Helmsay, welcome. Then from Vienna, the SEM and the Academy of Sciences that allowed us to kind of make this room and this location realistic. And particularly, we had today the session on the EU pilot projects. So these 6 EU pilot projects have come to a close, or are coming to a close within the next 3 months. And the HCA organoid that I'm coordinating has co-funded this meeting quite significantly, and the others have also contributed to the cost of the meeting. So thank you for all the sponsors that have made it possible to have good food and a nice ceiling and all the things that contribute to having an in-person meeting, plus obviously the high cost and effort of the virtual thing. Actually, the virtual, all the IT stuff is as expensive, perhaps even more expensive than doing it in person. So that was kind of really my lesson learned, that the virtual part is really hard to do right, obviously. And I would also like to thank the program committee who picked the speakers and designed the program and made a lot of contributions to making this meeting possible in the current form. All the speakers and breakout session leaders, you can see from the many pictures how diverse contribution or how many people have actually contributed here. And the people who have chaired the sessions, the microphone runners and scribes and people have contributed.
Yeah, so should I just take. So I want to echo Christoph's thanks to the microphone runner team here from his institute. Was amazing. And they worked incredibly well also with the planning team. And it's really a huge thank you to the international integrated planning team from SEM, the semis, so to speak, the UK and US staff that made this all come together. And in fact, the staff here, the audiovisual staff, and as Christoph said, this is all going online and the technical support from that. You may not have noticed all these people, but we have, you've noticed Sam and Jenny who've taken the online talks. We've got Christine and Tracy back there with also Kate and the semi support that I can recognize here. Anna Ewan and Anna Schwendinger and Pedro Martins and I'm mixing up people from different countries, but what I suggest is that we just give them all a huge round of applause. And I also want to take this opportunity to say thank you to Christoph, who's So just this is kind of where the reveal will come after this, but you've heard that there will be a development meeting coming up. There's also regional meetings in the next months, in October in Latin America and in November in HCA Asia, and then in Paris we have the HCA Developmental and Pediatric meeting. So you're all warmly welcome. Watch this space. There will also be online participation as well as in person. Again, that's the HCA tradition. And you see that the registration is already live for the pediatric and development meeting. And without further ado,
turn to me. So the only remaining thing is asking ourselves, where will we be next in 2023? And so we're very happy to say that next time we're going back to the United States. It's actually been a long time since we were in the West Coast. Our last HCA meeting in the Bay Area was February 2017. That was a long time ago. And so for further details, our new host, Jonah Kuhl.
All right, so this has been an incredible meeting, and I'm very, very happy to announce on behalf of Steve Quake and myself and BioHub CZI and the San Francisco Bay Area HCA community that we will be hosting next year in San Francisco, or thereabouts. And the tradition of kind of ending one meeting with a teaser and hype for the next meeting is kind of my honor and distinction, so I get to be the hype man here. And I think this is a tall meeting to follow. Like the bar that you set, Christoph, is impressive. I can guarantee that the ceiling will not be nearly as ornate. Very unlikely that the weather is as warm. San Francisco has great food. So, we may do okay in the food category, but this has been great, and next year, I think is gonna be really wonderful, maybe just to kind of emphasize the amount of progress that has been made since everyone was in the Bay Area 6-ish years ago, and kind of looking forward to next year and the progress that will continue to be made, and we can follow up on the integration of these atlases and continued efforts throughout the community, hopefully while also having a really fun time in bridges and mountains and an HCA tower. We'll see if we can convince the Salesforce people to light up HCA. I thought, one can dream, keeping with the tradition of my family home. Yeah, I think it'll be great, and obviously the vibes of San Francisco, there may be even some purple unicorns and flower power and all the good things that San Francisco has to offer. You never know, you never know. Okay.
And you're back to me. Okay, so next time we'll be in California. Come and enjoy the lovely weather and the beautiful views. In the meantime, there's one thank you that we haven't fully made yet, which is the thank you to all of you just for being so much fun and engaging and entertaining. And I don't think in 2016, when there were like 90 people at the Wellcome Trust, we really imagined what this community would be. And there's nothing more gratifying than being part of this community. Now, the amazing team that already got a lot of kudos and has done so much for us also arranged two last things for us to do. They have arranged a survey that you will take. And if you don't take, they will remind you to take the survey because they are so cool, awesome, efficient, and know how to do so many different things. And they have arranged for us a wonderful closing reception. So now you will all go and enjoy yourselves there. And see you again in 3 more meetings in 2022 at the very least. That's without counting all the BioNetworks weekly seminars and so on. And back in California in 2023.